Okay, good morning. So, online people, can you hear me? Yes. Online students, good morning. Can you hear me? Yeah. Everyone in the
Recording in
online, I mean,
progress.
in the Zoom, you have all access to the tune that we are discussing today, right? Sort of the practical tune, right? I have uploaded it to a Google Drive. This is also for you, right? Everyone is in the group, right? ISM 27. No, I learned. Right. So in the description, there's a Google Drive link. Right. I'll be updating that Google Drive and all the tunes, papers, and special seminar, whatever practical session, all the tunes are there. Okay. On a level, you can go and download if you have misplaced or if you want to. Sort of, it's accessible to you all the time. මට කරදර කරන්න එපා. Right. I'll be uploading it, updating it, and online people, everyone has got it, right? Anyone new? Brand new. Okay. So this is the practical session for today, team. Okay. Right. දහම් පාසල් යනවා කියලා කට්ටියක් missing නේද? So this is the main tune that we have actually sort of at the end of that tune. Okay. With the help of your friends, and I also send you the recordings. You can complete it. Okay. Pass. You haven't got the tune. Okay. I'll put it in the Zoom chat as well. Then hold on. New, right? Okay. So this is the practical session today, and so the main one. Okay. You
ඒක
can go through
අක්කා
the
කියලා තියෙන
recordings
හොයන්න.
and complete it. Okay.
එන්සයිම්ස් කරාද? ඒක තේරෙන්නේ නැහැ. උගන්නන්නත් බැහැ. මෙයා ඉස්සරහට ගිහලා නේ කියන්නේ ක්ලාසේ. ඊයේ බෝඩ් එකට ඇවිල්ලා එයාට විතරක් කියන්න බලන්න ලියන දෙයක් කියවෙන්නත් බැහැ.
Online students, the tutor is available in the Zoom chat as well. Okay, right. Let's wait another maybe 5 minutes. Then I can start. You can hear me and see me, right? I got a new tripod there. So today I actually sort of am jumping a little forward. Ne? So we were at the end of cellular structures, ne? Right? So the section that we have to actually start is cell division today.
තෙලියුණ
Right?
පාලන කරගන්නේ
But
ෂෝක් එකට.
I thought of having this sort of special seminar, sort of
මේ සර් ගන්නේ.
just to keep your interest. Maybe once a month, something interesting we have to do. And also, cell before moving on to cell division, I thought having the introduction to metabolism is kind of sort of a different thing, right? Because under enzymes, you have a lot of
අද ෂෝට්
assay potential,
නෝට් එකකුත් තියෙනවා.
right? Assay potential එක ගොඩක් තියෙනවා. Complete full assays අහන්න පුළුවන් from enzymes. So before moving on to maybe cell division, I thought of giving you a bit of a essay writing practice today as well, right? So we are going to have a essay writing session, and yeah, we'll try to cover enzymes within today, and also the introduction to metabolism as well. What is metabolism and different? Let's say we have to talk a little bit about ATP and how ATP is produced and things like that. Quickly, we'll cover that. Yeah. So that's the thing. You have to sort of tackle the syllabus in chunks, right? So you have to go to your head. The enzymes is just a small section in the syllabus. It's not nothing. It's not much, right? Once you have completed the entire syllabus, you will see it's a such a like not even 0.1% of the syllabus, right? But that's why, right? You have to be able to appreciate how big the syllabus is, and also chunks filling you have to cover the syllabus, right? ලස්සනට පිළිවෙලට lesson එක ගාන ගිහිල්ලා නෙවෙයි. You have to within one day if you are covering the enzymes, that's good, right? So you have moving fast towards covering the syllabus very fast, right? So that's what you need. Quickly cover the syllabus so you can focus on the other two subjects and keep doing questions for bio, right? Self study eකට focus කරන්න පුළුවන් if you have covered the syllabus quickly, right? Okay. Someone whose mic is unmuted. Now okay, right? Yeah. Right. I think we better start, no? So, for the new people, I always sort of refer the resource book, right? I don't give a note, no? Right? So, I want your resource book to be whatever the thing, when I'm saying ask you to sort of jump to the resource book, you have to have the resource book, right? So, you have to do the nice highlighting and maybe small things to write down in the resource book. But mainly, you have to write the things I say in the tute, right? So, for today, the tute is the small 3-page document I handed to you, right? Okay, let's start. So check out your resource books first. Okay, resource books first. Metabolism. Okay, online people, if you have any connection issue, doubts, immediately put it in the chat. Okay, right. So page number 48. Page number 48. Energy relationships in metabolic reactions, metabolic processes. So that's sort of where we start for today because we are starting metabolism today. Page number 48. Everyone. So metabolism. So in the second lesson, so we have studied about the basic chemical molecules, right? Organic molecules that are involved in sort of making up organisms, right? Carbohydrates, proteins, nucleic acids. Then we went to the next level of organization, right? Subcellular structures. We talked about that. Now we are moving on how all those molecules interact with each other within the cells to do different processes inside the cell, right? So what is the umbrella term that we use to all the biochemical reactions taking place in a cell? That is metabolism, right? So first highlight page number 48. What is metabolism? Sum of all biochemical reactions of living being is known as metabolism, right? Sum of all biochemical reactions. Yeah. It consists of anabolic and catabolic reactions. So I have talked about this. If we talk about the umbrella term of metabolism, right? It includes everything, right? So it includes breakdown reactions and synthesis reactions, right? Anabolism and catabolism. Catabolism is like Cardano, right? Breakdown. So from complex molecules to simple, we are breaking down. And in this process, what's going to happen? Energy is going to be released. So this kind of reactions are called what? Catabolic reactions, exergonic. They are going to release energy. Yeah. Anabolism, නිකන් අනල හදනවා වගේ, right? Catabolism is breaking down, anabolism is synthesis. So it's the opposite of this. We are going to make complex compounds from simple to complex. So this process involves formation of bonds. So to create those bonds, what do we have to provide? We have to provide energy. So energy is absorbed, endogenic. Simple things, right? So highlight in the same page. Catabolism is breakdown of complex molecules into simple molecules by releasing free energy. Highlight, that's a definition, very commonly question. Right, very commonly question. Okay. Then it's an exergonic reaction, right? So catabolism is breakdown, releases energy. Okay. Anabolism is the opposite, is the making of complex molecules from simple molecules by absorbing free energy. So it's an endogenic reaction. So in your the tute for today, there's a space here in the first page. Metabolism killed. Under there, draw two arrows and quickly note this down. Online students, I'll be delivering this tute to you, right? In the next month's tute delivery package, this will be included. Okay, this tute will be included in the next month's delivery package. So here, we have a lot to cover today. Then we move on to whatever the way that we can either provide energy to do anabolism, or during anabolism how energy is released. That is in the form of our what? Universal energy carrier. Right? That is in the form of our universal energy carrier, that is sort of common to sort of all living systems. All living systems, adenosine triphosphate is sort of the universal way of giving energy or taking energy, right? Usually there are derivatives, right? With GTP කියලා කෙනෙක් ඉන්නවා, right? So in some systems there can be some other derivatives of ATP, but ATP is the most, is like, let's say, most widely like energy sort of currency that we can observe, right? So it's important that we study a little bit about ATP, because in future we are going to talk about photosynthesis and cellular respiration. These are all at the end of the day මේගොල්ලෝ ඔක්කොම try කරන්නේ to produce ATP, right? So before studying those processes, it's important that we know what ATP is, right? So next page, page number 49. ATP acts as the energy carrier in all living organisms, including the simplest bacteria. Therefore, ATP known as the universal currency of energy transactions. Okay, nothing to highlight, right? Those are just some sentences, right? Then next one highlight, they can ask you, energy can be defined as the capacity to do work. Now it's physics, right? Energy can be defined as the capacity to do work. Work is what? Force into distance. Force into distance. Yeah? Then work is what? Divided by, what is the equation for work? Work done. Okay. I have very happily given up physics from my life. Not happily. It was, I mean, it was okay while I did it. Yeah, okay, so energy is the capacity to do work. Right? You have to have energy if you want to do some work in any living system. So, what is the energy form that we use? ATP. All organisms require energy for their living processes. Such processes are, right? So, they can ask you to name few of the energy requiring processes. Right? මෙතන ඉන්නවා කීයද නැද්ද? 7 තියෙනවා. Right? At least 4 5 remember. Right? Remember the common ones. Synthesis of substances, that is called what? Anabolism. So for anabolism, you need ATP. Active transport across plasma membrane. We have talked about this, right? When we are talking about the plasma membrane, I have told you there are some transporters in the plasma membrane that require a payment, right? There are two kinds of transporters. There are just protein channels. ඔහේ protein channel එක through යන්න පුළුවන්. හැබැයි not anyone. It's like, what is the example I took? It's like at your school gate, there's a security guard. Only schools, like school children are allowed to go through. But do you have to pay him? Saligala then? No. So that's like a protein transporter, right? Selected things can go through the transporter, but there is no need to give energy, right? No need to make an energy payment. So if this is the bilayer and the sort of the channel protein is like this, right? So things can go through here without any payment, but only selected things. This is a channel transport. If you take an active transporter, they are sort of much more complicated in structure. One of the commonest active transporters is there's a transporter called sodium potassium cotransport, right? Sodium potassium cotransport. What it does is, actually it brings K plus inside and it takes Na plus outside, right? And the special thing about him is එයාට අපි ඇතුලෙන් මෙහෙම ATP payment එකක් දෙන්න ඕන, right? highway එකට enter වෙනකොට enter නෙවෙයි, so when you're going out of the highway you have to pay a toll, like that. Active transport. For active transport you require ATP. Then transmission of nerve impulses, you will learn about this, right? How nerves transport the signals is an energy requiring process, right? Then muscle contraction, also there's a separate theory for that, right? Called sliding filament theory. It's in your syllabus. We'll talk about that when we are at that point. Beating of cilia and flagella. You know, cilia and flagella are motor organs of the cell. Yes. What is the organization of cilia and flagella? Composed of what? What is the composition of cilia and flagella? Online people? Yes. මොනවද හැදිලා තියෙන්නේ cilia and flagella? Last week, right? The day before last. Microtubules. Microtubules in arranged in what way? Arranged in what form? Yes. 9 plus 9 plus 2. And at the connection point where they are connected to the cell, 9 plus 0. Ah, nice. මතකයි නේ. 9 plus 0 and 9 plus 2. So cilia are just smaller versions of flagella, right? So all those movements require ATP. Bioluminescence, it's a bit interesting phenomenon. Not everyone can do it. Give me an example for an animal that can do bioluminescence. What do you mean by bioluminescence? Production of visible light in living organisms, right? So very common animal that can do it. Firefly, yes. Firefly. Anyone? Just like I can't remember the English name. Rabadulla Kilin, right? They can't fly, but they have like a glowing back end as well, right? So that is like a very special metabolic process that can produce visible light, right? Okay, so that is bioluminescence. Electrical discharges, very common. Who can produce electrical discharges? Even we can at our nervous in our end of our neurons, we can produce small electrical discharge. In our brain, there's a lot of electrical activity, but we can't like give someone else a shock, right? Who can do that? Electric eel, no? Electric eels can do that. Viduliyanda කියලා කියන්නේ they can produce like a massive shock. That's how they sort of hunt their prey, right? They can paralyze using a electric current, right? So you can write in front electric eels. Yeah, eel. Electrical discharge. So those are some of the basic things that organisms use ATP energy for, right? So we are getting some unique things, right? But the synthesis of substances, active transport, ඔය වගේ දේවල් everyone does it, right? යටට යන්න bioluminescence ඕගොල්ලෝ කරන්නේ. So those are unique things, but those are also energy requiring things. Okay. So next we need to know how all these energy relationships are interconnected. Everything comes down to what? Object, right? All the energy relationships in the universe, I mean on earth, right? It all comes down to what source, what object. You can understand why ancient people thought it's a god, right? The sun. Everything comes down to the energy we receive from the sun, right? That is the source of all our energy forms, whether it's mechanical energy, chemical energy, electrical energy, or whatever. You can trace the source to where? To the sun, right? So that's why next in your resource we have, and also in your tube, I have given a small energy flow diagram, right? Energy flows into biological systems from the environment through solar radiation, right? So sun is the start. Okay. Then what is the next step? Who can capture this, right? Now humans can somewhat has sort of we can capture it and convert it into electrical energy using solar panels, right? But who made the solar panels? Humans. Humans are powered by what? Food. Who made the food? Probably an animal or a plant, but the animal ate a plant as well. So ultimately, where did we get that energy to make the solar panels? From the sun. So you can trace everything to the sun in that case. So energy flow is into biological systems through solar radiation. So highlight primary energy source is the sun. Okay, primary energy source is the sun. Online people, follow the resource book. Okay. Then light energy is captured in the cells having photosynthetic pigments, chlorophyll, by the process of photosynthesis, stored as chemical energy in organic compounds such as carbohydrates. Okay, so immediate conversion, no? Light energy is converted to chemical energy by photosynthesis and then stored, right? So stored as chemical energy highlight as carbohydrates, right? Stored as carbohydrates. What is the storage form of carbohydrate in plants? Yes, online people. What is the storage form of carbohydrate? Very commonly, storage. It is converted to glucose කියලා ටියුට් එකේ තියෙනවා. Yes. Glucose goes another conversion. Starch. Why are we not storing food as glucose but as starch? Some general knowledge. What are the properties of glucose compared to starch? Glucose is water soluble. That is the biggest problem, right? Plant it's a water system, right? So if you are trying to store something that is water soluble inside the plant, ඇවිල්ලා බලද්දී ඒක නෑ ගිහිල්ලා වතුරට ගහගෙන ගිහිල්ලා වගේ, right? But if you store starch, starch is not water soluble, so you can keep it somewhere, it will stay there. That's why we are using starch. Okay. So compounds such as carbohydrates, within brackets write down in the resource if you like, starch. Then captured energy in organic food is transformed into chemical energy by a process called cellular respiration, right? This starch will have to be broken down to glucose. Okay, so we have to digest that starch to glucose first, right? So we are going to do a small experiment regarding that starch. We'll just so up to now, what is the process? Udimina sun, then photosynthetic organism, we got what? Glucose. Very commonly, right? There are other substrates. This glucose was converted to starch. This starch will be broken down to again glucose, then glucose to ATP, which is the universal energy currency. We can use this for anything, right? So this part happens in photosynthetic organisms. This part happens in photosynthetic organisms. This is in cellular respiration, right? If you like, you can write this down in the tube, right? Here, energy flow in the biosphere. Yes, glucose is soluble. Yeah. Kuta, please send to the everyone chat, okay? To the public chat. Personal chats, I tend to miss them, okay? Yeah. So it's a really simple process, right? Photosynthetic organisms are very commonly what? Very commonly plants, but there are other organisms. Cyanobacteria, right? There are bacteria that can do photosynthesis. And there are organisms under protozoa called planktons. Planktons actually contribute a lot to the photosynthesis. They are floating in the ocean, right? Kilometers ganang, there are like floating, let's say, masses of plankton. Whales, they eat those plankton, and those planktons, even though we can't see, they contribute a lot to production of oxygen as well as production of this chemical energy, right? But what we can see is plants, and we commonly eat plants, so that's why we are considering them as the main primary producers, right? Okay. So with that, we can move on to a little bit of detail about our actual ATP, adenosine triphosphate. Any questions up till now? No. Right. ATP, adenosine triphosphate, is what? It's a nucleotide, right? We talked about nucleotides. You have to be able to draw the simple structure of ATP, okay? You have to be able to draw the simple structure of ATP. What are the components of a nucleotide? තුනයි තියෙන්නේ වැඩි දෙයක් නෑ මතක තියාගන්න. What are the components? There's a sugar. What kind of a sugar? Pentose sugar. What is the pentose sugar in ATP? It's ribose. Alright. What is the next component? What करके? Yes, there's a nitrogenous base. So in ATP, what is the nitrogenous base? Adenosine triphosphate, right? So adenine. හතරයි නේ linen, guanine, thymine, cytosine. ඔය හතර නේ. So in ATP it's adenine. What is the remaining part? It is triphosphate. So we have to have 3 phosphate groups. Adenosine triphosphate. Right? So in the tube, quickly draw this. You have to be able to draw this one and do the labeling as well. Manage the space a little bit, okay? Yeah, any question? Online people, any question? You can see the board, right? I bought some new marker pens for you. Some pen hai. Hey, look at the tips, massive ones. So you can say this is the actual ATP, right? If you take from this point, then it's a what? It's a ADP, right? So this Kone phosphate or the terminal phosphate is the important one, okay? Online people, you can complete the tube once you have received it, okay? It will be in the next tube package that will be delivered to you. All the tubes are in the Google Drive, online people, in the group description, okay? මට කරදර කරන්න එපා. Tubes ටික තියෙනවා in the Google Drive. Okay? Right. So this is ATP, Universal Energy Currency. Come to the resource book, page number 50. Come to the resource book, page number 50. Most biological reactions use the energy released during breakdown of terminal phosphate bond. Therefore, it can carry energy anywhere. So, sorry, ATP is mobile. Therefore, it can carry energy to anywhere in the cell for any energy consuming reaction. So, ATP is like money, no? You can spend the money and get any work done very easily anywhere in the cell. But what's the problem with money? If you keep it somewhere for a bit of extended time, there's a chance that you might lose it, right? Because it's energy dense, right? I mean, it can easily release energy. So there's a chance that someone else might use it. We cannot store the ATP inside the cell. So we have to produce ATP with the demand. Right? ATP it's not a storage compound. It's a mobile compound. So we have to produce ATP with the demand. Right? Vadipura ATP hadanna bෑ. And also those are enzyme regulated process. Vadipura ATP hadenne right? My point is you cannot store ATP, right? Because it's very mobile and can easily break down and release energy. ATP can be produced within living cells within a short period of time using ADP, inorganic phosphate, and energy. Production of ATP within cells is called phosphorylation. Highlight. Production of ATP within cells is called phosphorylation. So, what are the components we need to usually to make ATP? ADP plus inorganic phosphate. We usually write with pi, right? It will give ATP. But we have to provide energy for this reaction. We have to provide energy. Then we can get ATP. Write down this reaction underneath this molecule quickly. ADP plus pi ATP. Simple things, right? Production of ATP within cells is called phosphorylation. Highlight that definition as well as write it down in the tute. Production of ATP within cells is called phosphorylation. Why? We are adding a phosphate, right? Because we are adding a phosphate to the ADP, it's a phosphorylation. Okay, simple things. Next, we have to talk about the several ways living cells produce ATP or how they several ways of phosphorylation we need to know. Okay. So there are three kinds of phosphorylations that is in your syllabus, right? Photophosphorylation, substrate phosphorylation or substrate level phosphorylation, and oxidative phosphorylation. Three things. Okay. Three things. Come to the resource book. So, photophosphorylation is what? Highlight the definition. Synthesis of ATP using solar energy in photosynthesis. Highlight, online people also. Synthesis of ATP using solar energy. Here, no? This conversion is photophosphorylation. Substrate phosphorylation. Synthesis of ATP using energy release by the breaking down of complex molecule into simple ones. Then I erased it. What is that called? Release of energy by breaking down complex molecule into simple. Catabolism, right? In front of it, right? Catabolic reaction, right? Catabolism. Yes. Catabolism. Finally, oxidative phosphorylation. Highlight the definition. Synthesis of ATP using energy released as a result of oxidation of molecules. Oxidation of molecules. What is oxidation? Right. There's a certain entity, right? It's releasing an electron. That is called oxidation, right? There's someone else to accept that electron. But this process, this electron is a high energy electron, right? This electron is a high energy electron. Now, when we are giving it from a high energy state to a low energy state, this electron is going to release energy. That energy we can harvest. That is what we are doing in oxidative phosphorylation. Okay, we'll talk about this in a lot of detail when we are doing cellular respiration. That's the basic idea, right? In a dam, right? In a hydropower generating dam, what do? We store water at a higher place. No? Then we open the gates, the water is going to come out, and we can use that to what? To generate electricity. It's like this as well. We are going to release high energy electrons from a higher place to a lower place. So when they are coming down, they are releasing energy, and you can use that energy to produce ATP, right? Exactly like a hydropower dam. You will see it's like there's some rotating turbine inside cells. Okay, that is powered by uh, sort of not electrons, right? But it's like that. It's like a dam like a hydropower generator right? It's actually a rotating structure inside the cell. Okay, clear about how we can generate ATP? What are the three types? Yes, photophosphorylation, substrate level or substrate phosphorylation, and oxidative phosphorylation. Okay. So final two, substrate and oxidative, you can see both in cellular respiration. Okay. The excited electron is fallen to the ground state to one. So who is giving the electron? It's called the what? If someone is giving an electron, it's called what? It's getting oxidized, right? If someone is receiving, it's getting reduced, right? So this one is getting oxidized and releasing electron. This one is accepting, right? Those are things where did we learn in reducing sugars. We had that story, right? Electrons accept karla, reducing sugars. Okay, so then we can come to the sort of the final part, right? Different uses of ATP. It's the same sort of the work, different tasks that were done by energy that has been now sort of given around this sort of diagram. Page number 51, right? So ATP can be used to produce light in bioluminescence, to produce heat in maintaining body temperature, to produce electricity, right, in those electric eels, then mechanical energy, muscle contraction, chemical energy in the synthesis, right, in the anabolic reactions, right? So those are the many functions of ATP that we can see. Okay. I have given some diagrams about how about this active transport at the end of the page. First page at the end, I have given how an active transporter works, right? You have to make an ATP payment and then the substance will be transported. Then second one, I have showed you like these microtubules act as railway tracks. Cytoskeleton has microtubules, right? So they are like railway tracks inside the cell. Motor proteins can sort of, with a load on their back, sort of walk along the microtubules carrying a load. It's like a, let's say, like an internal railway system that these transport proteins, also called as motor proteins, they can carry different things inside the cell along these microtubule tracks. That is, they are also powered by ATP, right? They are also powered by ATP. Any questions regarding this introduction part? Right? Any questions? Yes. And also, I forgot to mention here, you need to know the exact value of, it's given, right? Yes. We need to know the exact value of this bond breakdown, right? So either breakdown or making of ATP, it either, so if you're making ATP, it absorbs this energy. If you're breaking down ATP, it releases this, right? What is the value? Minus. So if you are releasing some energy, it's in the form of minus, no? What is that value? minus 30.5. You need to remember this value, minus 30.5 kilojoules per mole. Mole එක දන්නවනේ, Avogadro constant or something. ATP molecules mole එකක් break down කරොත් you can get 30.5 kilojoules or 30,500 joules. Yeah. Yes. So a lot of energy in one mole. There's a lot of energy. You need to remember this value. Okay. So if you want to make ATP, you have to give this. So it will become a plus. Yes. ATP breakdown, it will become minus. Remember this value. Okay. Let's quickly do an MCQ. So the printed one you have is sort of there's a small error. If you go to the very last page, there are a few questions, but the same MCQ is given twice. That is a small error I forgot to correct before sending it off to be printed. So sorry about that. Mhm. So there's this different MCQ I have included. Can you cut one of those questions? So I think the same MCQ is asked twice, right? Can you cut one of those and write down this one? I'll erase the board. Online people, you can anyway see the question, yes? Because online people, I have sent you the updated tune, right? The one I made the changes to. But ඔයගොල්ලන්ට තමයි වැරදි එක හම්බුනේ. Okay. Please write this question down. ATP. Yes. Please write this question down. ATP. Write this question down and please try it. ATP is a nucleoside containing pentose, adenine, and phosphate can be produced. Okay. People also try this question. Following statements regarding enzymes is correct. So cut it, cut one of them, yes, and write down this. Million people send me the answer. Send me the answer. Okay. What is your answer? Tama කරනවද? So even though you don't know what is pyruvate oxidation, you know this one. Yeah. Ah, yeah. That statement is a bit tricky, no? Statement number 4 is a bit tricky. I'll give you a hint. It's wrong. You don't know what is pyruvate oxidation? It's wrong. Maybe there's a better answer, no? Than that you can simply get an answer, right? So that's why it's very tricky to classify MCQs. You can understand, right? Try because immediately if you have after you have learned something, if you do something active such as doing some questions, they are going to go into your sort of proper understanding because knowledge without application is useless. Yeah, mekaning illa mekaning elite, right? So you have to apply knowledge immediately. Immediately apply. Yeah. Okay. Yeah. Yeah. What's the answer? Yeah. Yeah. Online people. What's the answer? Anyone who's saying 3? Okay. Any other answers? Okay. Let's discuss. Right. ATP is a nucleoside containing pentose sugar adenine phosphate groups? No. What is a nucleoside? Without a phosphate group, right? So ATP is sort of very opposite that it has 3 phosphates. So definitely not a nucleoside. Ne? Nucleoside එකක් වෙන්න බෑ definitely. So what is a nucleoside? You can write down it's a nucleotide without phosphate, right? So that's how we sort of review. Okay, you have to write, sort of learn from MCQs, write reviews of your own. Make it මොකක්ද වැරද්ද? Write down. So when you come across this question again, so අනිවාර්යෙන් ඔයගොල්ලෝ before going to the exam, you're going to do the full papers, so, and you're going to come across this MCQ because these are new syllabus past paper questions. Then, if you have at least written the answer down as a review once before, you know that MCQ point already. So it's a nucleotide containing pentose sugar, adenine, and phosphate groups. Okay, can be produced by oxidative phosphorylation using solar energy. It can be produced using solar energy, yes. But is it oxidative? Yes. Then what phosphorylation? Photo. Okay. Then you can cut this, right? The correct one. Photophosphorylation using solar energy. Next statement, hydrolyzes to ADP releasing 30.5. Yes. Ready on that? ATP reaction ATP goes to ADP plus inorganic phosphate releasing what? 30.5. So why do we write it as minus? You will learn about this when you are doing, there's a lesson called energetics for chemistry. Alright. So you will understand how why we put plus and minus in different reactions. Right. Enthalpy, entropy කියලා ලොකෝට ඔහොම cycle අඳින්න තියෙනවා in chemistry. Okay. Which lesson are you at right now? කොයි lesson එකේද ඉන්නේ? Structure and bonding. You are not doing chemical calculation at the same time. Samara class will karna. Then the next lesson is fourth one is gas laws. Then energetics. Pahatamai here talking about energy relationships in reactions. Okay. The next one. Formed in pyruvate oxidation through substrate level phosphorylation. That's wrong. Okay. Pyruvate oxidation is part of cellular respiration. Contains deoxyribose. What is the pentose sugar in ATP? Ribose. So correct it. It's ribose. It's ribose. Okay. That's all about the introduction to metabolism. Any questions? We can move on to the sort of the day's work, that is enzymes, right? Any questions? Online people? No, no issues. Going too fast? No. Right? Simple things, right? Just 2 pages within 1 hour is, I think, too slow. Okay. Right? Any question? Okay. So, enzymes. Page number 51. Enzymes. So, if you are in a biological field, right, any field related to biology, the importance of enzymes is vast, right? Because in living systems, almost all the reactions are enzyme catalyzed, right? Nikang ohe tibbata reactions wenna in a living system, we need to give it some enzymes. Because why? Usually living systems are at a, let's say, compared to, I mean, the temperature required for chemical reactions, our body temperatures are actually at a low level, right? So for most chemical reactions, you need a higher temperature. You need a high temperature. Temperature is sort of a measurement of how much energy is there. But because we are living and because of the what denaturation sort of behavior of proteins, a bit high temperature එකක් දෙන්න බෑ. We cannot provide a very high temperature in our body because our proteins are going to be destroyed, right? So because of that, from the very first stages of the early cells, right, from the first days of the very first cells were formed, there was a requirement to have enzymes inside cells. නැත්නම් reactions ටික කරගෙන විදියක් නැහැ, right? So because of that demand, enzymes evolved. So proteins කිව්වා, okay, we'll become this sort of promoters of reactions, so we can do those reactions, we can live and survive, right? So some proteins evolved to be what? Catalytic proteins. So catalytic proteins are enzymes, right? That is the importance. So page number 51, highlight what is the definition of an enzyme. It's a macromolecule which acts as a biological catalyst. It's a biological catalyst. So usually enzymes have what level of protein organization can you see in enzymes? Protein organizations mataka primary, secondary, tertiary, quaternary. So enzymes start at what level? Tertiary level there are quaternary protein enzymes as well, but at least it has to be tertiary level, right? So if you take a sort of a normal simple enzyme, මේ වගේ alpha helix regions තියෙනවා, beta pleated sheet regions තියෙනවා, ආපහු helix regions තියෙනවා, ආපහු මේව pleated sheet ගියපු regions තියෙනවා. So it's a globular protein. It appears in the sort of a clump of polypeptide chain arranged into different secondary structure organizations, right? So that's why they say it's a globular protein. Okay, different interactions. So can you remember disulfide bonds, Van der Waal interactions, hydrogen bonds? Or okay, K bonds, this has been stabilized, right? That's why it remains as a globular protein. You get it? But if we put some excessive heat energy to this, it will become denatured. Then it's not functional, useless. So today we are going to sort of demonstrate that how heating is going to destroy enzymes. So first property, there are 10 properties in page number 51. Right? First property, they are a globular protein. Then again, I have to teach you a little bit about energetics, because in the second statement they say they are biological catalysts. They lower the activation of the reaction they catalyze, or they increase the rate of the reaction. No. These are actually chemical kinetics, right? So if you take the direction of a reaction, it's all chemistry, right? What are the two things in a reaction? Reaction reactants do something together and produce what? Products. So in a reaction, we have reactants and products. Right? So in any reaction, if the reaction goes forward, that means the reactants are indeed producing the products. There's an energy hurdle, right? Energy jump for these reactants to get across. In a reaction, it's not an easy thing. They have to jump an energy hurdle, right? So that energy hurdle is called activation energy, right? So we have to give them some extra push for these reactants to become products. That is called activation energy. So if we draw sort of the curve for this reaction, there's a hurdle or a, let's say, a energy barrier for these reactants to jump. That is called what? Yes, we write it EA, or activation energy. Okay. You will see that now, because this and this, so the y-axis is free energy. Okay, so it is indicating of free energy. This is the direction of the reaction, right? Progress. The the reactants are having more energy than the products, right? So what does that mean? They have released energy, because the reactants had more energy and they have become less energy products. So they have released energy. So මෙතෙන්ට මෙන්න මේ gap එකට කියනවා අපි enthalpy එක කියලා, delta G කියලා. ඒක ඔයගලන්ට ඕනේ, right? ඒක chemistry වලදී අහගන්න. So that is not what we are concerned with. We need catalyst to get to jump this hurdle. Which hurdle එක උසයි. Very difficult. So if we are, let's say, if we are doing a reaction in the lab, how can we jump this hurdle? If we are doing a reaction, we can heat it, right? Usually by providing heat, we can jump this hurdle. But in the living system, as I told you, we cannot provide a lot of heat. ප්‍රශ්න වෙනවානේ, denature වෙනවා, රිදෙනවා. Right. So that's why enzymes evolve to reduce the height of this hurdle. මේ hurdle එකේ height එක අඩු කරන තමයි we got enzymes, right? So if we have enzymes, so this is the activation energy without enzymes. Okay, this is EA without enzymes. Without an enzyme. If we introduce an enzyme to this reaction, this hurdle will actually get reduced like this. Right? Now we have a different activation energy. We call it EA2. That is with enzyme. පයින් ඕන තාප්පේ පොඩ්ඩක් උස අඩු කරා වගේ, right? So we have reduced the hurdle height. So the reaction has a higher chance of going ahead. That is what the enzyme is doing. It is reducing the activation energy. Yes, clear. That's what the enzyme is doing. So there's a higher chance of reactants becoming products. So in your tute Puta, right? This page, relationship between activation energy and enzyme. There's a small topic here, right? යට ඒකට යටින් ලියා ගන්න ඇඳ ගන්න this here, okay? Relationship between activation energy and enzyme. If you like, you can write down this is the sort of the energy change, right? Delta G එක, right? This can happen in the opposite way also. This is for something additional. මෙහෙම වෙන්නත් පුළුවන්, right? The products can have more energy than the reactants also, right? මෙහෙම වෙන්නත්, right? So reactants and products can go like this as well. කොහොම කරත් there's an activation energy hurdle to jump. Got it? Products වල වැඩිපුර energy තිබ්බත් there's an activation energy hurdle you need to jump. So without the enzyme, this activation energy hurdle will be tall. So this is with the enzyme. Alright. This sort of reaction is what? This is an endogonic reaction. Products are having more energy. Make one. This is the one in your resource. No? You need to be able to draw this. Free energy, progress of the reaction. Higher one is at activation energy without the enzyme. Shorter one is with the enzyme. Also, you can write EA1, call this EA1, okay, activation energy 1 is greater than EA2. EA2 is what? The activation energy with enzyme, which is low. Chemistry you have to draw what is called mono deviation curve, something like that. No? Yes, sir. You have to then indicate this is the sort of how the reaction works without the enzyme and how with the enzyme it sort of increases the proportion. Oh my god, in chemical kinetics you will learn about that. Any question? No. So we are at still at the second property. Okay, page number 51. We are still at the second property. That is, enzymes reduce what? The activation energy, thereby increasing the rate of reaction. Okay. Too fast? Right. Alright. Okay. Third property. Most enzymes are heat labile slash sensitive. Right. Because why? Enzymes are proteins. Proteins can be denatured. Like that. Or a globular structure. The nice functional unique 3-dimensional blah blah. It will be broken down into something non-functional. Usually to the primary structure level. After denaturation, the protein can be broken down. So that structure is functionally useless, right? Functionally useless. It cannot do any function, because from what level onwards do proteins have functions? Tertiary level onwards. So if you have gone back to either primary or secondary, that protein is functionally bit useless. Get it? So that's why we say they are heat labile or heat sensitive. We'll see how heat sensitive they are today. Okay, all right. Then fourth one, their presence does not alter the nature or properties of the end products of any reaction. Right? We have an enzyme in a reaction කියලා, there is no difference in the reaction that is happening. For example, if you are talking about breakdown of starch to what? Glucose. Starch to glucose. What is the enzyme usually involved? Amylase. Amylase enzyme. Hitia කියලා, this starch to glucose breakdown, the products will not be altered. It is the same glucose that we know. There is no difference in the glucose produced, right? So it doesn't matter if it's an enzyme catalyzed or not, the reaction is the same. What is just happening? The activation is reduced, activation energy is reduced, so the rate has increased. Okay. Then 5. Okay. Before coming to 5, read number 6. Most enzyme catalyzed reactions are reversible now. Most enzyme catalyzed reactions are reversible. Starch to glucose as well as glucose to starch you can do. Yes. Because the thing is, the enzyme is actually catalyzing both reactions. Do you get it? Right? Maker, you will understand there's the final lesson in final 10th lesson is called equilibrium in chemistry. Right? Equilibrium. This sort of reactions you study. Right? When you introduce an enzyme here, actually the forward reaction as well as the backward reaction is catalyzed, right? But because the forward reaction is more favored, that is happening in more proportion. Do you get it? Right? Both the forward reaction and the backward reaction are having their activation energies reduced. Can you see? So reactants and products is the forward way, right? So products to reactants is the backward way, right? So if you want to convert a product to a reactant, you have to also jump back. Yeah, faster pine on, right? So faster pine it has been made easier, because to jump backwards also you need to get across this energy hurdle. Activation energy can pine. So yes, forward reaction, reactant to products has been made easier, but at the same time products to reactants has also been made easier, right? Can you see? ඉස්සරහට පයින්නවා නම් this hurdle පස්සට පයින්නවා නම් almost the same jump you have to make, right? That's why we say almost all enzyme catalyzed reactions are reversible, and also they catalyze both forward and backward reaction, right? But because the reaction is progressing in the forward direction, we usually get the products. අපිට නිකන් products ටික ඩඩක් ගාලා ආපහු reactants වලට convert වෙන්නේ නැහැ. Because why? In this given moment, the reaction is progressing forward. You will understand this when you are doing this equilibrium lesson in chemistry. Okay, got it? Okay. Then come back to let's leave say property number 5. Property number 5 එක පොඩ්ඩක් තියලා ඊළඟ එකට jump කරන 7. The rate of enzyme activity is affected by pH, temperature, substrate concentration and inhibitors. මේ ඔක්කොම ගැන අපි ඉගෙන ගන්නවා. Right? How temperature affects, right? How the pH affects, how substrate concentration affects, everything we are going to learn. Then next one, they are not being used up during the reaction. This is interesting. Okay. Shall I erase this? This one. You're done, right? Online people? Anyone who needs this? I'll take a photo and put it here. That one. Want to make work here, right? Okay. They are not being used up. Because why? They are not a reactant to be used up. මොකක් reaction එක involve වෙන්න එපා to be used up. Are they a reactant? No. They are just there to push. ඒගොල්ලෝ reaction එකේ involve වුණා කියලා they are not being used in the reaction. Right? They are just there to push. Okay. So that's an important property that we need to sort of let's say understand, right? Okay. So if we take this reaction starch, right? We have to add to starch what? Our enzyme. Let's say starch plus amylase is here. There's going to be a middle, let's say a transitional stage. Mad stage එකක් තියෙනවා. It's an unstable complex that is sort of formed by the enzyme and the what? We call this as the substrate that the enzyme is acting on, right? So there's a middle stage that we call as enzyme substrate complex, right? මැද මෙහෙම stage එකක් තියෙනවා. Then from that onwards the reaction will progress to what? What is the product? Glucose. But the interesting thing is what? The amylase is going to be here as well, right? එයා වෙනස් වෙන්නේ. Amylase is going to be here as well. Alright. So there's a reactant and enzyme, and the middle there's an enzyme substrate complex, and you have a product enzyme. So the enzyme can again come back here and do the reaction all over again. You get it? So this is what? This is our substrate, starch. Glucose is what? Our product. Okay. Write this down. Underneath mechanism of enzyme action, write this reaction. Online people, there's a space here under these two diagrams. There's some space for you to write some blah blah. Write down there. Enzyme substrate complex, and this is what? This is the enzyme. Okay, this is the enzyme. Here at the end also you have the enzyme. All right, people, any question? Okay. So you are not clear about this enzyme substrate complex. Actually, if I draw this kinetic thing again, right? These are the reactants, these are the products, right? මෙහෙමනේ curve එක යන්නේ, right? This is the reactant, this is the product. At this point, where the maximum energy is sort of the highest energy point, you have this complex. This is the place you have that complex. Enzyme substrate complex එක මෙහෙම උඩට නැගලා පල්ලම් බහිනවා to be the product, right? Didn't give it to you because it's not in your resource, but that's the logic. Okay, because of this complex, it's difficult to make and it's also very unstable. It's unstable. Okay? Yeah, වැඩි වෙලා හැමින් it will fall down to be the product and the enzyme. The difficult part is this climb. මේ උඩට climb එක තමයි is the difficult part. But once they have made this high, reach this highest energy point, they will fall back down to produce the product, right? So this enzyme substrate complex is a bit unstable intermediate. Okay, you don't need to know about, because without the chemistry knowledge, after you have gained the chemistry knowledge, this will start to make sense. Okay. So once this reaction is complete, amylase will go and bind to another starch and do the reaction again. Amylase is very quickly doing the reaction, right? It's because it's not being used up. He can do so many reactions, right? Clear. Okay. Then the other things, they are not being used up. They possess active sites. I'll talk about those later. Then some enzymes see nitrogenous components to catalyze the reaction, which are known as cofactors. All those will come across along the lesson, right? So that is basically the intro to enzymes. What is the definition of an enzyme? It's a macromolecule which acts as a biological catalyst, right? That you should memorize, right? Okay. Page number 52. Moving on to what? The mechanisms of energy reaction. Sorry, enzyme action. Mechanisms of enzyme action. Few terms you need to know. Few terms you need to know. Okay. First one, the reactant on which the enzyme acts on is referred to as the substrate. Highlight very first sentence. The reactant on which the enzyme acts is referred to as the substrate. So in this reaction that we took, what is the substrate? Starch. So highlight, this is a definition. Reactant on which the enzyme acts on is referred to as a substrate. The enzyme binds to the substrate, forming the enzyme substrate complex. While the enzyme substrate complex form their enzyme and the substrate form their complex, the catalytic action of the enzyme converts the substrate to the product. So we have enzyme plus substrate forming the enzyme substrate complex, and at the end you get a product and the enzyme again. Enzyme unchanged. Okay. The reaction catalyzed by each enzyme is very specific. The specificity of the enzyme results from its shape. Then next definition. The substrate binds to a specific region of the enzyme. This region is called the active site. Highlight what is active site. Highlight what is active site. Board is a bit unclear. You mean my handwriting is a bit too small? I need to flatten these markers. Right? So what is active site? the substrate binds to the specific region of the enzyme. So I'll in the tube you have, okay. Can you see there's an enzyme that is given? So the substrate, can you see there's a green one and a orange one that are coming near the enzyme, right? So the purple color enzyme. Can you see there's like a pocket in the enzyme for these substrates to enter? Can you see? There's a pocket. Into that pocket, the substrates are nicely fitting, or building blocks, Legos, right? So they are nicely fitting. So once these substrates are bonded to the active site, the enzyme reaction is going to happen and convert the substrate to the products. That's the basic story, right? So you can see that diagram in the tube, right? That is basically what happens in any enzyme catalyzed reaction, right? So the substrate comes, binds to the active site, then forms enzyme substrate complex, which is unstable. Then it's going to result in the product and the enzyme. So the products will be released. Simple story. The active site is formed by a few amino acids, and other amino acids are needed to maintain the shape of the enzyme molecule. Right. So when we talk about how enzymes work, there are actually two theories, right? Those two theories are, because in your resource it's a bit messy, right? How they have given නිකන් මෙහෙම වුණාට මෙහෙමයි but මෙහෙමත් වෙනවා වගේ එකක් තමයි තියෙන්නේ, right? So there are two mechanisms of enzyme action. Mechanisms of enzyme action as lock and key hypothesis, lock and key hypothesis, and induced fit. ඔය hypothesis දෙකේ නිකන් jumble එකක් තමයි they have given in your resource as under the mechanism of enzyme action. Induced fit hypothesis, right? So if I explain the lock and key hypothesis, it's like this. Imagine the enzyme as a door lock, right? So that door lock will only be unlocked, or that door lock will only take its own specific key, no? වෙන අතුරු දාලා කෙරකෙව්වට හරියන්නේ නැහැ, right? So lock and key hypothesis states that the active site is a very rigid place. The lock it's made of metal, right? අපිට වෙන දේවල් දාලා කරකවන්න බෑ. So they say the active site of an enzyme is a very rigid structure or a part, and it can only be bonded to the substrate that is specific to it, right? The substrate is like the key and the lock is like what? The enzyme active site. You get it? The enzyme active site we can only insert its own substrate. So that's how they say in the lock and key hypothesis the specificity is given. What do you mean by specificity? One enzyme only reacts with one substrate. So amylase only reacts with starch. So why? So under the lock and key hypothesis, they say that is because of their shapes, right? Because of the shape of the active site of the amylase or whatever enzyme, no one else other than starch can fit into it, because it's like a lock and a key. That is what they mean by lock and key hypothesis, right? So in your tute, I have given the diagram for the lock and key hypothesis. Can you see the first diagram? This one, top. Underneath it to උඩ හරි කොයි හරි ලියන්න lock and key hypothesis, right? Lock and key hypothesis. Can you see the enzyme is the bigger one? Always the enzyme is bigger, you know this. So if I draw a rough diagram of it, එකනේ අර building blocks වගේ නේද? මෙහෙම. Right? And this is your substrate. Substrate එක nicely fits into the active site. So this is your substrate. This is your enzyme. Please label these things in the tube. And what is this? This sort of the interlocking place. What is it? Active site. Right? So this is the active site. වෙන කෙනෙක් ඔබන්න බෑනේ මෙතෙන්. The substrate, only the substrate has the proper shape. Right? So label this. Is it my connection or is it blurry? Is it blurry? All right. Please send in the chat. Is it blurry? No. No. Okay. From time to time. No. Okay. Maybe it's your connection. Right. Okay. Got it about the lock and key hypothesis. Let's write a little bit. Read. Write down there. There's some space. I should have put more space. Right. Write this down. Oh, may put my Attinglian star mark here. Right here. Put a dot or star mark or whatever and write this down. According to lock and key hypothesis, according to the lock and key hypothesis, the specificity of an enzyme, according to lock and key hypothesis, the specificity of an enzyme is attributed to the complementary shapes of the enzyme active site and the relevant substrate. Can I repeat? What did I say? According to lock and key hypothesis, the specificity of an enzyme is attributed to the complementary shape of the enzyme active site and the relevant substrate. Right. So lock and key කියන word එක it's not in your syllabus, but they talk about it, right? The specificity is due to the complementary shape, right? But now there is another theory, because with sort of further studies they have seen that not all enzymes are like this, right? There are some enzymes that are sort of, let's say, accommodating. Like they can sort of be, let's say they change. When the substrate is coming close, the sort of the enzyme slightly changes because I want to do this reaction, right? So that's what we talk about in induced fit, right? Lock and key, it's not very rigid, right? So that is sort of the most widely accepted hypothesis at the moment, the induced fit hypothesis, right? So under the second diagram, this one, yes, the yellow and the green thing, write down induced fit hypothesis. Induced fit hypothesis. Okay, induced fit. Then write down where underneath where you wrote down about lock and key, write down according to induced fit hypothesis. According to induced fit hypothesis, according to induced fit hypothesis, as enzymes are not rigid structures, the interactions between the substrate and the active site may slightly change. The shape of the active site so that the substrate and the active site become complementary to each other. Substrate and the active site become complementary to each other. So if I say it again, according to the induced fit hypothesis, as enzymes are not rigid structures, the interactions between substrate and the active site may slightly change the shape of the active site so that the substrate and the active site become complementary to each other. Okay. That is what happens most of the time, right? So there's an enzyme waiting and there's a substrate coming. Because of the sort of interactions, they sort of become, let's say, accommodating of each other, right? After they have married, they start to live with each other, right? And then they have to accommodate each other. Because why? They have to do the reaction. That is what the induced fit hypothesis says. Okay. Any questions? So those are the two main mechanisms of enzyme action. How enzymes work. So then keep reading. For any enzyme catalyzed reaction, it is very essential that the enzyme and the substrate has a very nice tight fit, right? There should be a nice tight fit. That's why we also, in both these hypotheses, there's a nice tight fit, right? In the lock and key, there's definitely a tight fit. In the induced fit also, because they are slightly changing their shape, they become complementary to each other nicely. Lassana tightly fitted. That is necessary because bonds have to be broken and new bonds have to be created. Open enzyme making some bonds are broken down and maybe new bonds are created, right? So to do that nicely, there should be a nice tight connection between the enzyme and the substrate, right? So keep reading the resource. The tight fit not only brings the substrate molecules and the active site close to each other, but also ensures the correct orientation of the molecules to help the reaction to proceed and to catalyze the conversion of the substrate to product, right? Those are things, just have an idea. Thereafter, the products depart from the active site of the enzyme, then the enzyme is free to take another substrate into the active site. So now enzyme is free, it can go and catalyze the reaction all over again. Simple. Any questions? That is mechanism of enzyme action. No, clearly. So can we do any questions? Mm, Yes, we can do MCQ, that is not in your tube. Is it in your tube? Hmm. I'll put it in the board, right? Let's try another question. This question, question number 7, which of the following is a characteristic of enzymes? You guys please write this down, okay? Online people, can you try? Question number 7, send me the answers. So ඉස්කෝලෙන් මොකද කියන්නේ 80% එහෙම හොඳට බලනවලුද කොහොමද මේ කාලේ පොඩි භීෂණයක් යනවනේ බය කරනවා නේද, right? At least 50% වත් තියාගන්න නැත්නම් ඉතින් parents අටවත් ගිහලා කතා කරන්න බෑනේ පොඩ්ඩක් 50% වත් නැත්. No, anyway, if school is not productive, don't go, right? But at this point, go. There's nothing for you to study much. නිකන් ලොකුවට workload එකක් නැහැ. So at this point, go and go to the physics lab and touch the traveling microscope or something and මොනවා හරි අල්ලලා බලන්න, like micrometer, screw gauge, ඔය වගේ ටිකක් touch a few things, even if they are not doing the practical, right? මැඩම්ලාට ෆෝස් කරනවා අපිට මේව ඕනේ නැහැ කියන්නේ නැහැ එපා, ප්‍රැක්ටිකල්ස් කරමුද කියලා ටිකක් ගොට්ටල්ලන්න. තියරි ඕනේ නැහැ මැඩම්. එතකොට ඒගොල්ලොත් සතුටින් ඔයගොල්ලොත් සතුටින්. Alright. Online people, try this question and send me the answer. Okay, good, good. I'm getting answers. Easy, no? Yeah. What's the answer? You know who's still doing? Okay. Okay, great. Let's discuss which of the following is a characteristic of enzymes. So they're asking the correct statement. They do not alter the nature of end products. Yeah. ඒගොල්ලෝ හිටියා කියලා ඒගොල්ලෝ there's no impact to the end product, no? Right? Whatever the reaction that they are just there to push, just to catalyze, just to lower the activation energy. ඒගොල්ලෝ හිටියා කියලා there's no issue. So that is the correct one. But let's sort of review the others. They increase the activation energy? No, they reduce the hurdle, no? So you can cut this and write. They reduce the activation energy. So the hurdle they have to jump is reduced. Reduce the activation energy. They are not substrate specific. No, they are very substrate specific, right? This is one of the things that we sort of use in pharmacology, right? Most of medicines are actually sort of very related to enzyme action, right? Most medicines are either enzyme inhibitors or activators, right? We are trying to stop in enzyme action or increase enzyme action by giving most of the medications, right? You will see some with the examples. Many medicines are either in sort of interfering with the enzyme action, right? So that is because if we sort of influence a particular enzyme, we can be sure that that particular reaction will be changed. That's because why? That enzyme is substrate specific. When a reaction should affect bayak. If you are targeting one enzyme, right, there is no risk that other things will be affected. Because why? The enzyme is very specific, right? Yeah, react only with the specific substrate only. Fourth one, a small amount of enzyme is used up during the reaction. No, enzyme is like it's not used up. Small negative product what useful. You can write any amount of the enzyme is not used up, right? Again, can you see it's like they have to trick you? Maybe chuttak use not done it there, right? So once you have covered the entire syllabus and go to the exam, maybe chuttak use not done there, right? So that's how they sort of try to play with your sort of weak memory, right? Then finally, any part of the enzyme molecule can catalyze a reaction. No, no. Hematantambe. What is the special place? Yes. Cold, but what is the place where the substrate binds and the reaction happens? Right. Okay. Then you can write underneath the reaction takes place at the active site of the enzyme. You can write the reaction takes place in the active site. හැමතැනම බෑ. Yes, active site. Okay. Correct. Then we can move on with the theory. Factors can කතා කරලා I'll give you a break. Okay. Alright. So next moving on. Cofactors. Right? Cofactors. All clear about mechanisms? We have two mechanisms, induce fit and lock and key. All those are referring to what? What is the basic concept that you need to know? The enzyme is specific to the substrate. Right? React that is the basic idea you need to have. So moving on to cofactors, definition, highlight page number 53. Non-proteinous components that are essential for the catalytic activities of certain enzymes are called cofactors. Okay. Non-proteinous components which are essential for the catalytic activities of certain enzymes. Alright, highlight. Okay. They are non-proteinous because the enzyme is a protein. So these other things are usually carbohydrate based, or it can be other things such as inorganic things such as metal ions. Right? There are different ways, different cofactors. So you need to know they bind to the enzymes in two ways. Some bind tightly and remain permanently, while others bind loosely and remain temporary. Like it's obvious, right? Tightly bind to permanently no. Yeah? Loosely bound cofactors are reversible under certain circumstances. So talking about cofactors, I have told you a little bit about this when we are doing. We talked about when you talk about nucleotides. I told you a little bit about. Na? So cofactors are non-proteinous. There are inorganic ones. Inorganic means they don't have a carbon involvement in their structure. Inorganic ones are there and organic ones are there. Organic cofactors has a special name called, yes, coenzymes. Coenzyme. Where is the enzyme? No. Right? Basic classification. Right? Inorganic cofactors and there are organic cofactors. Organic cofactors are called coenzymes. Usually vitamin derivatives, NAD plus, FAD plus, biotin is also a vitamin. Right? So vitamin B5, B7 biotin, I think. Right? So usually vitamins are coenzymes. They are usually required for certain reactions to happen. Right? නිකන් සමහර enzymes වලට පාළුයි. Right? ඒගොල්ලන්ට මගේ cofactor එක නැතුව reaction එක කරන්න බෑම කියලා. So those are there to sort of regulate everything, right? Because if you want to regulate the enzyme sort of action, on evolution එකෙන් it has been decided on මෙයාට coenzyme කෙනෙක් ඕන. Without the coenzyme මෙයා won't do the reaction. Right? It's like that. For example, if you take vitamin B6, pyridoxine, that is a coenzyme in hemoglobin production. Right? So with vitamin B6 deficiency, there can be problems with making hemoglobin. Right? So vitamin B6 is a coenzyme for hemoglobin production. It's like that. Right? So just remember certain derivatives of vitamins, NAD plus, FAD plus, and biotin are the examples you need to remember. Okay. Inorganic cofactors are usually metal ions, right? Zinc 2 plus, Fe2 plus, and copper 2 plus. All right. Write down this classification in the tube under cofactors. Please write down the definition under cofactors in the tube. Here, non-proteinous components which are essential for the catalytic activities of certain enzymes are called cofactors. Right? Then do this classification. Cofactors as organic and inorganic. Organic are called as coenzymes. Zinc 2 plus, Fe2 plus, copper 2 plus. And write down the other examples. Biotin, NAD plus, FAD plus. So there's a small problem with the practical today. What? Finding amylase. So how can we find amylase? If you can donate some amylase, we can do the reaction. I can give you a beaker, then you can collect during the interval. Then I can show you enzyme denaturation also. I have brought everything except amylase because it's expensive and this is not provided in small quantities. It's usually used in industries, right? Amylase is used in certain industries. Okay. Okay. Got it about cofactors? Non-proteinous components essential for the catalytic activities of certain enzymes. Then organic cofactors are called coenzymes. What are the two ways that cofactors bind? Tightly and permanently, loosely and temporarily, right? Those are the two ways. Any questions up till now? No questions. Shape, ne? Okay. Take a break. Exactly the hammer I'm giving the break today. Quickly come back, okay? We have to do another 50 minutes. Quickly take the break. And also, if you can collect some amylase during the break. What's पता नहीं इंटरनेट का है. In your schools, there's a practical for salivary amylase, right? So, if you can collect, then we can do the practical. Right. One of the other method I call the absent, right? No, not here, not here, right? Not here. I'll give a separate one for the boys. At least 50. No, no, I'm just kidding. Okay, take a break. Don't go spitting in the canteen, okay? So we can, I can show you denaturation and then starch reaction also. Okay. Okay. Online people take a break. 10:45. Online people, can you hear me? Any questions you want to clarify? So new students online. So this is a free session. That's why I sent you the Zoom link. But usually the recordings and the Zoom link is available in the website. That is Guru Taru Online. And once you have enrolled, the tube delivery will start. Right. So it will take about 1, 2, maybe maximum 3 working days for you to get. And from next month, there's a new tube package. You will also receive that. Okay. Any feedback that you want to sort of any changes in the class structure? So you're talking with other friends now. In a classes, what do they say is sort of the best thing about maybe they are going to other bio classes? Anything you want to add to this class? So Martina, because we can sort of anyway we can change the structure and sort of according to your sort of feedback, right? Anything? This way is good. That depends after the results come. Class on the yeah, let's see. Right. MILS එකතු කරනවද දන්නේ නැහැ නේ තුන්දෙනා. ස්පීකර් එකක් නැහැ මේ. Come on. Come. Ah, see? Yeah. I thought you were collecting MILS. Okay. Right, okay. Need a paper class. At this point, I mean, we are discussing a lot of questions in the class, right? And to be honest, with the amount of theory knowledge that you have, the amount of questions we can do is limited. Right? So, yes, I will have full paper writing sessions close to your term test. That is in almost 2 more months, right? I plan to have together with the other teachers in the institute as well. I plan to have a sort of program to prepare for you for the term test. Maybe full papers. Not full papers means you're not going to get a full A-level paper for the first term test. No, probably 2.5 hour paper, maximum 3 hours. About 20 MCQs, 2 essays, and maybe 2 structured essay paper. First term test. Full paper එක කහන තරම් දෙයක් නැහැ. මුළු resource එකම අහුවත් there won't be any questions to make 50 MCQs, right? So anyway, I plan to have a exam preparation few papers before the term test, right? So that will be sort of I'll decide the time with the institute and the other sort of the teachers, and let's decide on that. But you have time for to prepare for the term test. So your immediate challenge is to sort of prepare for the term test, right? So my sort of advice is go for all 6 term tests. Okay, එක term test එකක්වත් don't skip. School theory, school teaching අවුල් වුණත්, papers අවුල් වුණත්, papers වල ලකුණු දුන්නේ නැති වුණා, go to the school term test, right? Because so when you are preparing for the first term test, you are going to study now because you have a goal, right? Because you have to study for this exam. So you are going to study anyway. It's it's a challenge for you to solve. There's a goal in mind. So you're going to study for the first term test. Then for the second term test, you're going to study that section as well as something new. Right? So when you have prepared for 6 term test, these early lessons are going to be sort of like permanently etched into your memory because you have prepared for them 6 times. Right? So this chemical and cellular basis, you might be like, it's the way you are supposed to be, right? You have to be experts in this. I mean, you have to have them sort of word by word when you go to the exam, right? Because when you are going to the exam, you have going to have some fresh memory as well, right? Langadima karapu molecular micro ageva, you might have only had time to study them once or twice, right? So you have to close to the exam, you have to pay attention to those, right? So close to the exam, maybe a month or two before the exam, you cannot start to study cellular basis from the very beginning. But if you have at that point have studied for 6 term tests, definitely you have a very good, let's say, confidence about the second, third, fourth lessons, right? So that's the benefit. ඒගොල්ලෝ ලකුණු දුන්නේ නැති වුණා, discuss කරේ නැති වුණා, just go to the exam and write the paper. That's it, right? Okay, let's continue. So we have to move on. So we have seen what is the nature of enzymes, and we have talked about how enzymes work and what are the other external things that are required for enzyme action. Now we are going to see certain physical factors that can affect the rate of enzyme. Okay? So you know, it's a biological catalyst. Anyway, it's going to increase the rate of the reaction, right? But if we change sort of different factors that we are providing for the reaction, we can change the rate of the reaction itself, right? So in your syllabus, we have 4 such factors, that is temperature, pH, substrate concentration, and presence of inhibitors. Those 4, right? So we have to see how these individual factors are they going to increase, decrease, or what they are going to do to the enzyme rate. Online people, you can hear me and see me, right? Any question you can ask. So first one, temperature, right? So again, this is sort of physical chemistry, right? What do they say? Page number 53. Come to the resource, page number 53. Okay. Increase in temperature increases molecular motion. Therefore, the speed of the moving molecules of both enzymes as well as the substrate will be accelerated. This will enhance the colliding probability for both enzyme active sites and substrate molecules. More collision between enzyme active sites and substrate molecules generate greater chances for the reaction to occur. Collisions. Have you seen in the lottery board when they are like lotteries, ticket lottery prices, when they are sort of in those raffle draws, have you seen there's like balls flying everywhere in like a chamber, and there's like a tube that is going up and then sucking one ball out meter, like in those lottery ticket. So it's like that. So the substrate molecules are like inside our cells, substrate molecules are sort of moving very fast, and there's an enzyme somewhere that the enzyme is sort of moving also, right? So when the speed of those substrate as well as the enzyme molecules that are moving here and there increases, the chance of them hitting each other increases, right? It's like that. So if the substrate are those like balls moving here and there in those lottery raffle draws, rat and ticket, right? And there's a sort of a tube going up that is like the enzyme. So it is going to connect it one of the substrates and going to go down. When the speed of that increases, the chance of hitting that tube increases. So that's what happens in enzymatic reactions as well, right? So you need to know this. There are 4 graphs that you need to know under the 4 factors. That is, first one, temperature. If we draw the rate of reaction for temperature with increasing temperature, the rate of reaction, it is an exponential, right? So rate, these these graphs you need to draw, right? I mean, you should be able to draw temperature. So when we are increasing the temperature, what the molecular motion is going to increase, the probability of collision between the substrates and the enzyme is going to increase, therefore the rate is going to increase, right? It increases like this. Okay, a bit more. Let's add a bit more gradient, maybe, right? It's going to increase. But this is not all good news, because why? Temperature is a good thing, as well as quickly it can become a bad thing. Right? After a certain point, temperature is a very harmful thing, right? So keep reading. This can continue up to a certain temperature, after which there is a rapid decline in enzyme activity. This temperature is referred to as the optimum temperature. This may vary from organism to organism, right? So with increasing temperature, the rate of reaction increases only up to a certain maximum point. It is called what? It is called what? Yes, optimum temperature. So that is the best temperature, right? We have the highest rate of reaction. Okay. So talking about this optimum temperature, organisms are designed to maintain this optimum temperature in their body. So there are warm-blooded animals, there are cold-blooded animals and warm-blooded animals. So after reptiles are cold-blooded. Birds and mammals, we are warm-blooded animals. Right? We have warm-blooded. What do you mean warm-blooded? Whatever happens outside, if we are not dead, we try to maintain the same constant temperature in our bodies. If we are healthy, right? Warm-blooded animals, we are going to maintain the same temperature in the body, right? Warm-blooded. But reptiles, amphibians, and fish, whatever, they can change their body temperature according to their outside temperature, right? But we, as mammals and birds, we are going to try really hard to maintain our body temperature. So when it's outside is very cold, what are we going to do? We are going to shiver, right? And also our skin is going to be very pale, because we are not letting blood come out to the skin. So if blood comes to the skin, we can lose heat, right? So our skin becomes very pale, no? Because the blood is not coming to the skin. And if we are very hot, we are going to sweat, right? And we are going to feel a thirst, and so everything is done to maintain body temperature. Our body temperature is not a random value. 38 degrees Celsius is our body temperature. It's our body temperature. If you are not feverish. So, is it a random value? This 38 degrees Celsius, right? Kau decide kara, ඔයගොල්ලන්ගේ temperature 38. No, it is also the optimum temperature for our enzymes. It is the optimum temperature for our body enzymes, right? That is because at 38 degrees Celsius, most of the enzymes in our body are at their optimum temperature. So they are providing the highest rate of reaction possible. So our body is very efficient. So through evolution, අපි මාළුන් ඉඳලා amphibians ළඟ ඉඳලා reptiles ළඟ ඉඳලා birds ඉඳලා mammals. So mammals are the most evolutionary advanced, right? So mammals having warm blood is a very advanced feature, because we can do our reactions at optimum temperature all the time. The මාළුවෙක් නම්, because their temperature changes when their body is very cold, sometimes their enzymes are not at the optimum temperature. They might be not very efficient, but that's okay, because they are not very advanced, right? We have to process a lot of brain power, right? And we are metabolically active, right? Most of our energy is actually consumed by our brains, right? So our enzymes have to be tip top, right? They have to be operating at their maximum all the time. That's why we have a constant body temperature, and also our enzymes are maintained at that optimum temperature. Got it? So examples, they say most of the human enzymes have optimum temperature around the body temperature, that is about 35 to 40 degrees Celsius. 35 to 40. human. Okay, but there's something interesting happening. So something additional. What happens when you get a fever? I wanna have it. Why do you get a fever? Because there's some problem in your body, right? Mocker infection usually due to an infection, right? Bacteria virus. So what is your body trying to do to get rid of that, whatever the invasion damaging thing? So that means your white blood cells are going there and they are making these antibodies and they are killing these viruses or bacteria. There's a lot of things happening, right? So that means the metabolism is high. So actually, they have given you a range, right? About 35 to 40. So if you take human enzymes, right, our actual optimum temperature can increase to about 40 degrees Celsius. අපි දුවන්නේ 38 වල වුණාට our enzymes can increase their rate up to 40 degrees Celsius. So you are having a fever, that means a fever around 101, 101, 102ට තියෙනවා කියලා කියන්නේ. So still that means the rate of reaction can be increased up to that. Right? Up to 38 we are maintaining it because going near 40 is kind of risky, right? There's a chance of something bad happening, I'll tell you, right? What is going to happen if you exceed the optimum temperature. But in a fever, it's a like a critical situation. We have to get rid of, we have to protect our body. So what the body does is, it's going to increase the body temperature in an attempt to increase the rate of reaction even slightly. Now චුට්ටකින් හරි rate එක වැඩි කළා, maybe we can increase the metabolism and fight this pathogen. That's why the body is giving you a fever, right? That's why the body is giving you a fever. Okay, so then ඉස්සර වගේ අර ice pack දාලා එහෙම නැත්නම් fever එක අඩු කරන්න try කරන එක, it's not the best. The body would be like, I'm increasing my temperature to survive and you're putting ice packs to absorb that heat, right? So the best thing is to take a painkiller and to properly hydrate, ne? හොඳට වතුර fluids බීලා උණ හැදුනහම එහෙම ඉන්නෝ, right? But our sort of belief is that උණ අඩු කරන්න ice තියන්න හරි whatever, like the cold water තවන්න හරි මොකක් කරේ, the body would be like, I'm increasing my temperature and they are trying to cool it down, ne? I'm trying to do something here, right? So that's the purpose, right? In a fever, the body is trying to increase the optimum temperature slightly so that the reaction rate can be increased. Okay. And the next example is, optimum temperature of bacteria in hot springs is about 70. That means that's their habitat, no? ඒගොල්ලෝ ජීවත් වෙන්නේ hot springs වල නම් they have to have enzymes that are optimum at that high temperature, about 70, right? That is no need to explain, no? Right. Then the next important thing is, what is going to happen when the temperature increases beyond this optimum temperature? What is going to happen? The hydrogen bonds, ionic and other weak chemical bonds of the enzyme active sites may get disrupted. This will result in a change of the shape of the active site of the enzyme, which will alter the complementary nature of the active site enzyme molecules. Therefore, the complementary binding of the enzyme active sites and substrate molecules will be prevented. This is denaturation. Like any other protein, enzymes can be denatured, right? Usually, it is an irreversible process. Once an enzyme has been destroyed or denatured, you can't turn it back. So that is what's going to happen after you have exceeded the optimum temperature. The rate of reaction is going to come down. At a certain point, the rate of reaction, it will not actually be zero, because without the enzyme the reaction can happen, but it will not be zero. In your resource it's given it is reaching zero, right? Then it's fine, right? You can understand, like enzyme එක නැතුව reaction එක වෙන්න පුළුවන්, බැරි කමක් නැහැ, right? It will not be zero. There will be some amount of reaction happening, but let's assume without the enzyme the reaction will stop here. Okay, so this graph you need to be able to draw, right? How? So this is a bit exponential at this point. මේක මෙහෙම ආවට චුට්ටක් near to the optimum temperature, it is going to increase a bit rapidly. Right? It's going to come like this and sort of like this. Okay. So in the tute, factors affecting rate of enzymatic reactions, put number 1 here, put number 1 here, temperature, and draw this. So if this is a human enzyme, put a value around 40 here, take this as 37 here, degree Celsius, 30, right? This is 30, 20, put numbers properly, okay? Right? Just before 40, you have 37, that is optimum temperature for human enzymes. Online people, any questions? So usually above 40 and 45, nearing to 50, human enzymes will be denatured, right? We cannot handle more temperatures more than 45, 50. Our enzymes are going to be denatured. So there's an amylase enzyme that we can extract from a bacteria that is called Bacillus licheniformis or something. That amylase is very resistant, right? We can heat it up to 100 degrees Celsius and it won't be denatured, right? So depending on the organism, they can have resistant enzymes, heat resistant enzymes, but not us, right? Because our body temperature is 37, 38, 39. Our enzymes are going to be denatured probably after 40, 45, like that. Okay, right. Moving on. Next factor. Oh yeah, then so they say the above event is called denaturation of enzyme molecules. Highlight what is denaturation in the resource. Page number 54. Denaturation of enzyme molecules. Therefore, the rate of enzyme catalyzed reaction will start to decline when the temperature increases beyond the optimum temperature and stops completely at a certain temperature, although the rate of collision will keep on increasing. Right? You get it, right? With the increasing temperature, the molecules are moving very fast, but there are no anymore enzymes. Enzymes have been denatured. If you right, if you think about it logically, if the collisions are happening, the reaction can happen. It cannot be zero, right? Zero in the bad, but we go with what your resource says. Enzyme is just what? It's just a catalyst. Help right? Without the enzyme, the reaction slightly can happen. But doesn't matter, right? What is in the resource. So say the it says stops completely at a certain temperature, right? So just write that. Although the collision rates keeps on increasing. Clear about temperature. Shall we write down a few things in the tute? Write down. Put a star mark and write optimum temperature. Put a star mark and write optimum temperature underneath this graph. Okay, make it the temperature at which maximum rate of reaction the maximum rate of reaction of a specific enzyme can be seen is called this optimum temperature. Right, okay. Shall we move on to the next factor? That is pH, right? Related to the acidity or the basicity of whatever the medium that this enzyme catalyzed reaction is taking place. pH, right? So that is again changes from enzyme to enzyme. But the interesting thing is your optimum temperature is sort of same within the same organism, right? If we take human enzymes, almost all our enzymes have the same optimum temperature. But if we take human enzymes, they can have several pH values where they work their best. That is because based on their location, where they are located or secreted or where they are working in our body, they can have different pH values where they are working the best, right? So that's a special thing. If you have the optimum temperature in the human body, almost all enzymes have the same optimum temperature. But when it comes to pH, they can have different pH values. Okay, that's the sort of the difference that you need to know. So read page number 54. Enzymes function most efficiently within a certain pH range despite maintaining the temperature of the environment constant. Right? So we are providing optimum temperature, assume, but the next factor that is going to determine the rate of reaction is the pH. Okay. So highlight the next one. The narrow range of pH in which a particular enzyme catalyzed reaction takes place is named the pH range. pH range. So what is this pH scale, from what number to what number are we drawing it from? What is the sort of with the scale, pH scale from 0 to 14, no? Usually 14 pH scale. So pH range for different enzymes, you have a pH range that it will work. I'm going to need a lot of space, right? So there are no enzymes sort of behaving at this pH 14, right? In the human body, basic nature, there are no enzymes, right? So I'll draw up to 10 and 11 maybe, right? So this is sort of the pH that we can see in our body, from maybe very acidic to slightly basic. There are certain places in our body, right? So different enzymes can have different pH ranges. That is the range of pH that they are working. That is the pH range. So the next definition is what? Highlight the pH at which the highest rate of reaction occurs is the optimum pH. So similar to optimum temperature, we now have optimum pH. Highlight. They can ask you what is optimum pH. Highlight. pH at which the highest rate of reaction occurs. Okay, keep reading. Alteration of pH above or below the optimum pH may lead to decline in enzymatic activity. This is due to alteration of chemical bonds involving the formation of enzyme substrate complex. In most enzymes, the optimum pH is 6 to 8. What is so special about this 6 to 8 pH range? 6 to 8 අතර මොකද තියෙන්නේ? Neutral pH is included in that. pH 7 is neutral, no? pH 7 is the neutral. So usually our body tries to maintain almost a neutral pH, but our blood pH is about 7.2. Okay, our blood is slightly basic, but very close to neutral, no? Right? So most human enzymes are sort of working in this pH, right? 7 to 8 range. That is their pH range. So what do they say? Going below this pH or above this pH is going to decrease the rate of enzymatic reaction. Okay. So a typical enzyme we can draw, if this is the maximum rate of enzymatic action, about 6 to 8 we can observe, with the peak around 7. This is what a typical human enzyme curve looks like. So what is this? pH range, and what is this value? Yes, optimum pH. Yes. So this is what like a normal human enzyme, this is a pH range. This is optimum pH. Usually where the maximum rate of reaction is, you can see, is the optimum pH. Simple things, right? But there are special places, such as in your stomach, right? Our stomach is like an internal acid vat එකක් වගේ, right? It's a very special place where we have a very low pH, about pH 2-3, right? pH 2-3 level it can reduce. So enzymes in our stomach can behave differently because they have a different optimum pH around pH 1 maximum pH. So they can have the optimum pH around pH 2. So pepsin is an enzyme that you can find in the stomach, right? Involved in protein digestion. So this could be pepsin, which is a stomach enzyme. It works best in very acidic conditions because our stomach secretes a lot of acid, no? Okay. Any questions? Pepsin works best as pH 2, and if we take like a normal small intestine enzyme such as trypsin, it works best around 8. Or I mean, I drew this not for trypsin, for a normal any other enzyme which works around neutral pH, right? So if you are drawing for trypsin, you can draw with the center around 8. Okay, you can draw with the center around 8, right? So this could be trypsin. Trypsin is an enzyme that you can see in the small intestine. So what happens is, when you have eaten something and so they come down the esophagus, right? Right? From your mouth to the stomach, there's a tube, and that tube is connected to your stomach. Your stomach is a bag of acid. There is acid. So here you have pepsin. Pepsin is going to do some protein digestion, and at the end of the small intestine, stomach, what do you have? Small intestine. So within tap connect small intestine. It goes like this, and there are it's very long small intestine. Okay. So at this junction, there's a valve, and methane elliot water. We are going to add neutralizing substance that is mostly bicarbonate. Right? HCO3 dala methionine में HCl we are going to neutralize. Okay. So this acidity is only limited to the stomach. Because it's very damaging. Your stomach has to go through a lot of suffering because it is a bag of acid. Right? The stomach has to generate a new layer every 3 days, I think. Every 3 days it has to generate a new layer because this acid is burning the stomach. Right? So that is why stomach enzymes have their optimum pH around at that very low pH value. But here, that point onwards in the small intestine, the enzymes can behave at the normal pH range about 7 or 8 because the bicarbonate is going to neutralize this acid and protect. Okay. So this yes, this stomach acidity is very beneficial as well as it leads to a lot of diseases, right? Because we are eating a lot of garbage, right? That they are not causing us a lot of damage because of our acidic stomach. This acid is going to actually destroy most of the bacteria that we eat, right? Most of the bacteria that we eat are actually getting destroyed in our stomach. pH 2 තියෙන පඩි acid එකක් දාලා burn කරනවා. So there's a like an immune function as well. You learn about this. Acidity in the stomach is an immune function, right? But at the same time, acid is acid, right? If the acid comes beyond here, methionine උඩට આવොත්, what do you call that? We call it gastroesophageal reflux disease. Right? Acid එක උඩට ගහනවා. But the problem is the stomach is protected from this acid, but not this esophagus and මේ එතනින් උඩ තියෙන්නේ මොනවද? You have your mouth and your pharynx and vocal cords over it, right? So this acid can burn everything above that, right? So that's a problem. That is gastric reflux, and it can cause damage to the esophagus, and long term it can cause cancers, even esophageal cancers, right? So that's something additional we'll talk about, that it's in under digestion. Okay. So what I was talking about was based on where the enzyme works in the human body, they can have different optimum pH values. Got it? Put number 2, write pH and draw this. Online people, if you can't see anything, immediately let me know. Okay. I think what do you suggest? Shall I give another attempt to find amylase and do the practical on another day? Or let's do it on another day. I'll try to find some proper amylase. Or I can send you a YouTube video and a beer. watch this YouTube video. There are so many Indian YouTube videos of doing enzyme denaturation, and different. So the other practical is we can change the temperatures and see how the reaction, I mean, rate of enzyme reaction changes. And we are supposed to do the experiment as 5 degrees Celsius, 10, 15, 20, 35, and if we do the practical properly, we would get this graph, right? We are going to get this graph. After we have heated the enzyme beyond 40, we'll be able to see the rate is dropping. Okay, that's what the proper experiment is supposed to be. Two things, it's going to take a lot of time because usually amylase takes about 30 minutes to work, right? So let's see. Okay, clear about pH? So usually due to this pH, what is the most I mean mostly affected type of bond, do you think? What kind of bond is affected the most due to this pH changes in the sort of the tertiary structure of the enzyme? Hydrogen bonds, right? In any denaturation, the peptide bonds don't break, right? The primary structure stays the same. Amino acid chain doesn't break usually. It's very difficult to break, right? What is going to change is the globular shape held together by weak disulfide bond, ionic bond, hydrogen bonds, van der Waal, homothene. ඔය ටික තමයි break වෙන්නේ. Usually it's very difficult to break the peptide bonds. Okay. But anyway, if the shape changes, what is going to happen? The shape of the active site changes. එතකොට induced fit හරි මොකෙන් හරි ඇවිල්ලා substrate it cannot bind. If it's not binding, the reaction is not happening. Get it? Right. Okay. Still drawing. Online people, any questions? So I'm supposed to be at the hospital today, but I'm doing classes and the sort of the group is going. Consultant has come, so I won't be marked. I will be marked absent today. So if he finds out I'm doing classes, repeat. Right. Okay. Do this. So usually, I'm at a surgery ward these days, and we have, in the hospital there's like a, let's say, like a system of wards are sort of. So if you are a patient and you're getting admitted one night to the hospital, at that night all the patients are going to be admitted to one ward, right? That ward is said to be having a casualty night. Okay, casualty night. So if you are having like a surgical problem like බඩ appendicitis එකක් ඇවිල්ලා එහෙම නැත්නම් අතක් කකුලක් කඩාගෙන එහෙම නැත්නම් ලේ වමන යනවා වගේ මොකක් හරි surgical case එකක් නම් you are going to be admitted to the ward having the casualty. So last night was our casualty ward, right? Casualty night for our ward, and today morning there are patients even under the beds, right? So on the beds there are two patients, usually, and there are patients under the bed as well. So we have a separate file say called flow patients. We have to maintain a different file for flow patients, right? So consultant ඇවිල්ලා යට බලනවා මෙතන history ගත්තද කියලා, right? History ගත්තද examine කරාද කියලා. Flow patients ලා ඉන්නවා, corridor patients ලා ඉන්නවා, right? It's like that. But so the only downside to any government hospital is actually the crowding, and maybe the washrooms are a bit shit, right? And but the best, I mean, you cannot get that level of care in any other institution, right? So let's say if you are a patient in the ward, medical students are going to come early in the morning and they are going to take your history and examine, and they are going to manage. So they are going to ask you everything. They are going to ask everything from the patient. ගෙදර ඉන්න කවුද, ගෙදර බල්ලෝ ඉන්නවද, එලා වෙජිටේරියන්ද, ඊළඟට ළමයි කී දෙනෙක් කියලා, ළමයි හම්බුනේ සිසේරියන් එකකින්ද, ළමයින්ගේ වයස කීයද, ළමයින්ට මොන හරි ලෙඩ තියෙනවද, ඒවත් අහනවා, right? We have to ask social history, we have to ask, right? So it's a very low, there's a very low percentage of missing anything, right? So if we find out anything, we are going to tell our seniors. Then the house officer is going to come, sort of the resident of the ward. එයා ඇවිල්ලත් අහනවා short history එක. ඊළඟට එයා ඇවිල්ලත් ward round එකක් කරනවා. ඊළඟට අපි එයි ඔක්කොම යනවා, there's a registrar කියලා කෙනෙක් ඉන්නවා, a bit above, right? So the registrar is going to go ward round as well, right? ඊට පස්සේ sir එනවා, the consultant comes around 10, දැන් ඉන්න sir නම් 8:30 වෙද්දී, right? So we have to go be there by around 7. So then the consultant comes and the consultant goes the ward round, right? He is going to again do the examination and the management, everything, and the investigation මොනවාද කරේ ඒ ඔක්කොම බලනවා එයා. So sometimes the consultant comes in the evening as well. අවසරත් ඇවිල්ලා ward round එකක්, right? So the consultant is going to look at the patient at least once, and the registrar, that is the second in command, is going to look at the patient at least twice. House officer, that is sort of the junior doctor in the ward, is going to look at the patient at least 10 times a day, right? And the medical students are going to go through the patients at least once or twice, right? So that is the amount of care, but the thing is it's crowded, and sort of the cleanliness is a bit low. That is like directly proportional to the crowding, right? But that's the case. So that's a good system based on sort of the, let's say, medical education and medical occupation, it's very connected. So the consultant sometimes comes just to teach, right? He can go and do a private practice any way he likes, because he's the consultant. He has no one to challenge him. අවද ward round ගියාද කියලා කවුරුත් අහන්නේ direct ඇවිල්ලා. So he can do a private practice from early morning to the evening, but the consultants come to the ward and they go the ward round and they teach, because they are sort of in that loop. They learn that way, so they are going to teach that way. It's like that. So it's a very good system that is sort of we should try to maintain for the, let's say, foreseeable future, right? But the sad part is sort of, so when people are in the hospital, they are very sort of grateful, right? And like, and they remember what the hospital did for maybe 1 or 2 days, and when there's a strike for some critical issue, right? So all that gratitude goes out of the door when there's a small inconvenience, right? That is the sad part, right? They don't know how underpaid or overworked sort of the medical community is, not just the doctors, the nurses, right? They are so overworked and underpaid, and they take a lot of risks, right? A lot of infection chances. Patient la vata karagena inne, right? There's a chance of even getting very dangerous diseases, tuberculosis, herpes, syphilis, ඔයගේ ඕන දෙයක් හැදෙන්න පුළුවන්. Occupational වෙන කිසිම job එකක ඒ වගේ occupational hazard එකක් නැහැ නේ, right? For the salary that they are getting, they are taking a big risk. So just wanted to say that, right? Okay. Moving on to the next factor, substrate concentration. Substrate concentration, this is a bit of a obvious thing, right? So increasing the substrate concentration increases the probability of collision between the enzyme and the substrate molecules in the correct orientation. However, the enzyme molecules will be saturated after a particular concentration, therefore there will not be any further increase. Please highlight the word saturated after particular concentration. That section. Online people, if I draw here, would you be able to see? Yeah, yeah. Factor number 3, substrate concentration. What is the substrate? It is our reactant. It is our reactant, no? Right? That will become the product. But in enzymes, we give it a special name, the substrate. Okay? So if we draw that, if we give a lot of substrate, the rate will increase. That is obvious, right? So this is again rate of reaction. This is what I'll. This is substrate concentration. So usually write concentration within brackets. Okay, right? Substrate concentration. So you can increase the substrate concentration and the rate will increase like this. There are less increase, because why? More reactants you have, the rate is going to increase, right? When you are pressing the accelerator, more petrol is going, and the engine will sort of race or the rev higher, right? But the problem is, do you have unlimited enzymes? No. The amount of enzymes found in the cell or wherever is limited, right? So after you have increased the substrate concentration to a certain level, there will be a maximum rate of reaction reached, but beyond that point, the rate will remain the same. No matter how much you increase the substrate concentration, will remain the same. So you can label this as, so this is optimum rate, so this is saturation. Saturation. This concentration is what? Saturation concentration. So is it clear? It's like this. We have so much raw materials, we have run out of machines again, right? We have all the substrates even more than we need, but enzymes are over. Okay. Online people, any questions? Not sleeping, right? It's not raining. Yeah. If you have any question, you can ask. Okay, for this, now case after this, there is more than enough surface. We continue. There is no use of increasing the substrate concentration. Reaching the situation that's well, okay, clear about this saturation. Write down underneath that. Write down underneath that. Online people also write down. Okay, can you explain the part enzyme molecules being saturated? It's like this. So imagine you have a machine. So enzyme is like a machine that is going to do what? That is going to do a certain work. That is going to take a substrate and make a product. Right? It's like a machine. So imagine you are putting a lot of substrate into the machine, keep putting, but there's a capacity for that machine, right? So if the machine is completely fully like it's like being filled with whatever the reactant, however much you increase, you cannot increase the rate of reaction. You can draw the opposite of this, right? You can draw the same graph with what? Enzyme concentration. You can draw the same graph with enzyme concentration, rate and enzyme concentration. What is going to happen? So, similar graph you are going to get. Why? Now there are too many machines, right? After a certain point of increasing the enzyme concentration, right? What is going to be the limiting factor now? Substrates. Then substrate madhi, you get it, but we don't talk about this. Why? Because usually the enzymes are producing very small amounts. Right? Usually it's the enzymes that is the limiting factor. So here what has become the limiting factor? Enzyme. But here what is the limiting factor? Substrates. Usually there are so many substrates, right? Starch na vidya adupaduak. We are eating a lot, right? But what is the limiting? Is usually the enzyme because we are producing it in very small amounts. So I just gave it this for your understanding. It can go either way. Whatever the case, when something becomes limiting, no matter how much you increase the other factor, the rate is not going to increase. Anitya rate is going to stay the same. So you can, yeah, I will give you a small note. It will help you to understand. No need to draw this one, okay? It's not in your syllabus. Okay. Now, under this substrate concentration, usually enzymes are produced. Usually enzymes are produced in small numbers. Usually enzymes are produced in small numbers. Therefore, usually enzymes are produced in small numbers. Therefore, their availability is limited. Their availability is limited. Their availability is limited. When we increase substrate concentration, when we increase substrate concentration, when we increase substrate concentration, at a certain point, active sites active sites of all enzymes active sites of all enzymes active sites of all enzymes will be saturated active sites of all enzymes will be saturated and the rate reaction rate will remain constant. The reaction rate will remain constant. Got it? Substrate concentration. Concentrate maybe concentration. Okay. Next, it's a sort of a serious thing that you need to understand. That is, what are these inhibitors, right? So, I mean, as the name suggests, they are going to inhibit the enzymatic reaction. But මේගොල්ලන්ගේම opposites. There are activators as well, right? But in your syllabus, we are giving a more weight to enzyme inhibitors, right? So obviously, if there is an inhibitor, what is it going to do? It's going to reduce the rate of reaction, right? Inhibit right? So in your syllabus, we are focusing on inhibitors. So let's read enzyme inhibitors. What is the definition? Certain molecules or ions that selectively bind permanently or temporarily to the enzyme molecules and prevent them from forming the enzyme substrate complex. Highlight that. Molecules or ions selectively binding permanently or temporarily to the enzyme molecules and preventing them from forming enzyme substrate complex. Right. Okay. So they are either binding reversibly with weak interactions or binding irreversibly with covalent bonds. Right. Shall I erase this temperature? So inhibitors, they are of two types, as reversible and irreversible. Right. Samaravati, we can remove. Samaravati, once they have bonded, that is it. Right. So there are several ways we can classify enzymes, but let's first classify them based on their sort of what sort of binding they have. So enzymes you can classify as reversible, irreversible. Okay. Irreversible usually bind with what? They say with covalent bonds, right? We can't generally remove these covalent bonds, right? So usually many toxins that are binding to enzymes, I mean toxins such as cyanide, like that, they are irreversible inhibitors of enzymes. Sorry, enzyme inhibitors. Okay. Reversible, they bind with sort of weak interactions, and using different techniques, we can remove reversible inhibitors. Right? Right? Usually antibiotics are, I mean, generally there are irreversible ones as well. Usually antibiotics are reversible enzyme inhibitors. So antibiotics, what enzymes are they inhibiting? අපේවද bacteria ගෙවල. Bacteria ගෙව obviously, right? They are inhibiting the bacterial enzymes. So bacteria need different enzymes to make protein, digest nutrients. ඒගොල්ලන්ටත් ඕන enzymes අපි වගේ, right? And also they have like a cell wall. They have these, they require these enzymes to synthesize the cell wall as well, right? So for all these purposes, bacteria have enzymes as well. So if we can inhibit those enzymes using a drug, we can sort of kill those, right? Then we can sort of get better from the disease. That's why that according to that classification, enzyme inhibitors are of two types, as reversible and irreversible. Reversible are antibiotics, right? Certain antibiotics. Next classification of enzyme inhibitors, it's sort of the more widely sort of done classification. It's on how they how they behave, right? Based on how they behave. That is, they can be competitive or non-competitive. Competitive or non-competitive. This is more interesting. There is reversible and irreversible. Let's read about competitive inhibitors. Page number 55. Okay, let's read. Most of these are reversible. So you get it, right? So usually most competitive inhibitors are reversible. How do they behave? They resemble the shape and nature of the substrate. Therefore, they compete with the substrate selectively for the active site of certain enzymes. As a result, the number of active sites available for the substrate may decline, therefore reduces the rate of enzyme catalyzed reactions. The above situation may be reversed by increasing the substrate concentration. So we are having fake substrates here, right? Fake substrates. They look and behave like substrates, but they are not useful, right? They are not useful. What is their only purpose? Right? They are going to pretend to be the substrate and they are going to block the enzyme active site. But are they the substrate? No, they are just someone else. They are not going to produce the product that we need, right? What they are going to do is prevent the proper substrate from binding to the active site, right? They are competing. They are in the same playing field. They all look the same and they behave the same, but they are not the substrate. So when the substrate is going towards the active site, he has to compete with this sort of a competitive inhibitor because he is also going to the active site. Then another tarangyak, right? So this is what going to happen. What you need to remember is these competitive inhibitors are, they look and behave like the proper substrate, but they are not the proper substrate. They are useless, right? So what they are going to do is they are going to go and occupy the active site, preventing the proper substrate from binding to the active site. Okay, I'll show you this. There's a nice diagram. So if you imagine it like this, if this is the enzyme and there's an active site like this, right, that can accommodate this sort of a substrate, right? ඒ වගේ substrate එකකට active site එකක් තියෙනවා. So this is the proper substrate that is sort of like this. This is the proper substrate. This is the substrate. But there is a competitive inhibitor that is like this. So this one can fit in here as well. No? එයාටත් එතන fit වෙන්න පුළුවන්. Substrate එකත් එතන fit වෙන්න පුළුවන්. Can you see? So this active site can be occupied by this competitive inhibitor instead of the proper substrate. Yes. So substrate available නෝට අම්මට සිරි මෙතන වෙන කවුද ඉන්නවා right? So because they look the same. එයා කියනවා නෑ මමයි substrate එක enzyme ට තේරෙන්නේ නෑ මේක. Right? He thinks මට fit වෙනවා. So this must be the substrate කියලා හිතනවා. They are competing, right? So that is how competitive inhibitors work. So what can you do to reverse this? If there is a competitive inhibitor, what can be done to reduce its action? Online people, what can you do to reduce the action of competitive inhibitor? It's reversible, remember. Now set වුණාම ගලවන්න පුළුවන්. How do we reduce the action of this competitive inhibitor on this enzyme? Any ideas? We can increase this guy, you know. මෙයාගේ concentration එක වැඩි කරන්න පුළුවන්. එතකොට මෙයාව negligible level එකකට we can reduce. Substrate concentrations substrate concentrations ගොඩක් තියෙනවා නම් this competitive inhibitors effect would be reduced. Yes. Do you understand? So I'll give you an example. Do you know methanol? Right? Methanol is what? CH3OH ne? ඔගොල්ලෝ chemistry වලට තාම නෑ. Right? CH3OH is methanol that is found in මේ pen ink වල තියෙනවා, printing ink වල තියෙනවා, and in windscreen washer fluid කියලා තියෙනවා, wiper fluid ඒ වගේ තියෙනවා, and most commonly in Sri Lanka it's found in illegal alcohol. කසිප්පු වල තියෙනවා. Okay? How do we get alcohol? So we do some fermentation or sugar තියෙන මොනවා හරි දාලා ඒකට bacteria දාලා what do? We do something called distillation නේ. Distill කරන රත් කරලා we get the vapor and we cool down the vapor and we get the alcohol. Right? ඔයගලන්ට කියලා දෙයි how this distillation works. So what is the proper alcohol? C2H5OH නේද? දවසම මම chemistry කරා. What is this? C2H5OH is what? Ethanol. Ethanol ne අපිට ඕනේ වදින්න ethanol වලට, right? That is the addictive thing. We need ethanol. Yes, right? But when we are doing distillation, මුලින්ම එන්නේ මෙයා methanol because he is smaller, you will see, right? When you are doing chemistry, මුලින්ම එන්නේ methanol. හරියට දැන් ඉතින් ඔක දැන් කසිපු පෙරන මිනිස්සයාට he doesn't have like a degree in chemistry, no? Right? He doesn't know how to remove the methanol from the ethanol. හරිද? ඔය හරියට නිකන් ඔය හරියට ඔය වඩිය දානවා කියන like experts in කසිප්පු they know to let go of the first part of the vapor because they know this first part is poisonous. Okay, so what they do is they let go of the first part and then take the second part that is going to contain mostly ethanol. ඒකෙත් methanol තියෙන. Okay, so what is going to happen is this methanol is very toxic, right? Methanol is very toxic. And okay, what extra දෙයක් කියමුකෝ ඉතින් මම මෙහෙම extra දේවල් කියන්නෙත් නෑ. So this methanol CH3OH it's going to be broken down by the same enzyme that is breaking down ethanol. හැබැයි මේකෙන් හදන්නේ formaldehyde කියලා එකක්. Right? Formaldehyde. This formaldehyde is very toxic. Very, very toxic. Right? This ethanol breakdown to formaldehyde is done by the same enzyme that breaks down. So methanol breakdown to formaldehyde is done by the same enzyme that breaks down ethanol as well. අපි බිව්වහම අපේ liver එකේ ethanol breakdown වෙනවා. Right? Usually හැබැයි අපි ethanol breakdown කරොත් C2H5OH proper කසිප්පු ටිකක තියෙන්නේ එහෙම නැත්නම් ඕනම alcohol එක ethanol එකේ This is going to put acetaldehyde. Okay, wow, acetaldehyde is not poisonous. Yeah, okay. May are poisonous. Formaldehyde. But both these reactions are done by a same enzyme called alcohol dehydrogenase. Then, into these rural base hospitals, one of the most common presenting complaints is asippu bila ඉස වෙලා එනවා. මොකක්ද වෙලා තියෙන්නේ? මෙතනෝල් පොයිසන් වෙලානේ. මෙතනෝල් ටික බිව්වහම මොකද වෙන්නේ? What's going to happen? It's going to be broken down by the same enzyme, that is alcohol dehydrogenase, into formaldehyde. Very toxic, right? So what can you do? හිතන්න නිකන් ගමක ඉන්න පොඩි ඩොක්ටර් කෙනෙක් විදිහට ඉන්ටර්න් විදිහට ගිහිල්ලා ඉන්නවා. There's a case of illicit alcohol poisoning කියලානේ කියන්නේ. Illegal alcohol බීලා විස වෙලා එනවා. What can you do? There's a nice relationship between these two reactions. I told you, මෙතන ඉන්න common enzyme එක මොකක්ද? It's called alcohol dehydrogenase. අපි ප්‍රශ්නේ මොකක්ද? The methanol is being broken down to formaldehyde. Formaldehyde is very toxic. But this alcohol dehydrogenase can be competitively inhibited by proper ethanol. If we give some proper ethanol to that person, we can stop this reaction from happening. Methanol to formaldehyde reaction can be stopped by giving proper alcohol. Right? Right? Yeah, that's what you do. That's one of the emergency department treatments. Right? Methanol poison එකට ඒ කියන්නේ දැන් කසිපු බොන්න යන්නේ. බොන්න යන්න ඕනින්දනේ. බීපු මිනිස්සයාට තව බොන්න දෙන එක තමයි කරන්න තියෙන්නේ. Right? Methanol poison වෙලා යන මිනිස්සයාට අපි ethanol දෙනවා. Because why? Ethanol is going to competitively inhibit this reaction and do the proper reaction, stop methanol breakdown. Got it? And in Sri Lanka, that is illicit alcohol is the biggest thing, but in foreign countries, usually people drink methanol to suicide. Right? Or printing machines වල තියෙන ink cartridges තියෙනවා බීගෙන යනවා. Right? To suicide, because it's a sort of a potent poison. මැරෙන්න පුළුවන්. ලංකාවේ ඉතින් කසිප්පු බීලා illicit alcohol distill කරන්න ගිහිල්ලා තමයි methanol poison වෙලා එන්නේ. So do you understand? We can inhibit this reaction by giving his competitive inhibitor. Right? Competitive inhibitor එක දුන්නාම instead of methanol, it's going to react on ethanol. The same enzyme. Do you get it? Okay. Online people. Clear? Okay. Write down the topic inhibitors, factors affecting rate of enzymatic reaction, factors affecting rate of enzymatic reaction. Do this classification quickly, reversible and irreversible. Quickly. That's it. Don't draw this, okay? Not yet. No. So because competitive inhibition is reversible once the ethanol has been so we are giving ethanol lune, right? So once that person has drunk all the ethanol, they're asking if the methanol will start to work again. We are delaying the reaction. The methanol will be passed through urine, right? It will be passed through urine, but the risk is what? Methanol may risk it. Breakdown product. Formaldehyde is the problem, right? So we are delaying this reaction by giving ethanol. So the poisonous methanol that person has drunk is going to go out by urine. So we are delaying, right? So delaying until the methanol is passed through the urine. Okay. This is something bit too much. No, you got it, right? We are delaying the methanol breakdown until it can be passed by urine. Okay, right. Then something about okay, done. Let's talk about the other kind of inhibitor that is non-competitive. Yeah, Amutui, right? He is kind of dangerous than this competitive inhibition, right? I have given a diagram for that, right? Yeah, nikang amutuma sneaky kenek, right? So what this non-competitive inhibitor is going to do is he is not going to bind to the active site, right? If this is the enzyme, right, I drew earlier, he is not going to bind to the active site. So this is the active site. This is the active site, right? So if this is the active site. What this non-competitive inhibitor is going to bind is it's going to bind here to a site other than the active site. This is going to do a structural change in the active site of the enzyme. Structural change occur in the active site of the enzyme due to this binding of the non-competitive inhibitor. That is going to change this shape of the active site. The active site will be changed to something like this. All because this guy joined, right? You get it? I have put a diagram in the resource that you can understand. So you can see both competitive inhibition and non-competitive inhibition in the same diagram. Can you see? Online people? In the non-competitive inhibition, can you see there's a non-competitive inhibitor bound to a site other than the active site, and the active site has changed its shape. So now they are not complementary to each other, right? Complementary to each other. Read the resource book. Non-competitive inhibitors. They do not compete with substrate molecules. They interrupt enzymatic reaction by binding to a part of the enzyme other than the active site. This causes the enzyme molecule to change its shape in such a way that the active site becomes less effective for the formation of the enzyme substrate complex. Highlight, they cause the enzyme to change its shape. I don't need to give you two diagrams to draw, right? There's a diagram in the tube, no? Right? Okay. Now, draw this other classification. Enzyme inhibitors are of two types, as competitive and non-competitive as well. Quickly write that. Okay. So, okay, summarizing. What are the factors affecting rate of enzymatic reaction? First one, temperature, right? What is the temperature going to do? It's going to increase the speed of molecular motion. So, when the molecules are moving very fast, the probability of collision is going to increase. That is going to increase the rate of reaction up to a certain point. What is that point called? Optimum temperature. After that optimum temperature, what is going to happen to the enzyme if we keep on increasing the temperature? Enzyme denaturation until they say the reaction reaches zero, but collisions keep on happening. That is temperature. pH. So, most enzymes work within a narrow range of pH, that is called the pH range. And in that pH range, there is a pH at which maximum rate of enzymatic reaction is there. That is called what? Optimum pH. That can change according to the location of the enzyme and from organism to organism. Third one, what? Substrate concentration, right? Up to a certain substrate concentration, rate of reaction increases. After a certain substrate concentration, all enzyme active sites will be saturated with substrates. So, further rate of reaction will not increase. Finally, presence of inhibitors. Inhibitors always reduce the rate of reaction. Based on their binding, we can classify as them as reversible and irreversible. Reversible inhibitors you can remove because they bind with weak bonds. Irreversible you can't because they bind with covalent bonds. Then based on how they work, they are of two types, competitive and uncompetitive. Usually competitive inhibitors are reversible, right? Competitive inhibitors mimic the shape of the proper substrate, so they actively compete for the active site and they can be reversed by what? Increasing the proper substrate concentration. Non-competitive inhibitors do not compete for the active site, they bind to somewhere else and change the shape of the active site so the proper substrate can't bind there. Those are the four factors affecting rate of enzymatic reaction. Clear? Okay. Moving on to the final section that we have to cover today, right? Regulation of enzymes, right? Regulation of enzymatic activity. Okay. Mechanisms of regulating enzymatic activity. So basically, there's three you need to know called allosteric regulation, cooperativity, and feedback inhibition. I'll keep this and erase this. Right. So allosteric regulation is kind of a tricky concept that I mean to understand, right? So allosterism refers, I mean, it is very similar to what we talked about in non-competitive inhibition. Okay. So they say uh, allosteric regulation, page number 56. Let's read. In many cases, that the molecules that naturally regulate enzyme activity in a cell behave like reversible non-competitive inhibitors. Right? Usually, regulatory molecules behave like this. What? Reversible non-competitive. Okay? Please highlight that. They behave like reversible non-competitive. Is there a lag still? Lagging there? That is an important MCQ point, right? Usually allosteric molecules are behaving like reversible non-competitives. Okay. So regulatory molecules are either activators or inhibitors, bind to specific regulatory sites elsewhere other than the active site of the enzyme molecule via non-covalent interactions and affect the shape and function of the enzyme. Okay. So allosteric regulators are either activators or inhibitors. They can activate or inhibit. නිකන් regulate කරනවා කියලා කියන්නේ either you can increase or decrease. Got it? Allosteric regulators, they regulate, they can increase enzymatic action or inhibit. They can activate enzymes or they can inhibit. So both are done. Okay. They bind to sites other than the active site. Actually, it is called the allosteric site. Okay. The method, I didn't give a name, right? මෙන්න මේ අමුතු තැනකට bind වෙන්න කියලා I told you, but I didn't give the name. That site where these kind of regulatory molecules bind is called allosteric site. Huh? So they can be either activators or inhibitors. So coming to the allosteric activation and inhibition, most enzymes regulated by allosteric regulation are made of two or more subunits. Each subunit is composed of a polypeptide chain with its own active site, and the entire complex oscillates between two different shapes, one catalytically active and the other catalytically inactive. So these enzymes regulated by allosteric regulation are actually very complex, right? ඒගොල්ලෝ simple ඒවා නෙවෙයි. They are composed of either two or more individual protein sections. Those are called what? Subunits, right? They are composed of individual polypeptide chains, and they oscillate. We say, can you see in this diagram? In the top form, you have in the top part, the next page. Okay, this page, mechanism of regulation. Online people, refer the PDF. Can you see the enzyme is going between two forms as the active form and inactive form? Can you see? It's going between two forms. So take a guess and tell which form is going to do enzymatic action. Active form. Inactive form is useless. As an enzyme, it's useless. So what do you think? An activator, would it promote the formation of active form or inactive form? Active form, right? So activators are going to stabilize the active form of the enzyme. Can you see? In the first diagram, top part of the diagram, online people, there's a green color activator. I'll show in the screen also. There's a green color activator. When the green color activator binds, can you see? The enzyme becomes stabilized in the active form. එයාට inactive form එකට switch වෙන්න බෑ. Because why? The activator is in the middle, right? But if you go and look at the inactive form, there's a red color inhibitor binding there. Can you see? That red color inhibitor, what is it going to do? It stabilizes the inactive form, right? So inactive form is useless. This is how allosteric regulation works. Okay. So in the resource it says, when an activator binds with this regulatory site, it stabilizes the shape with functional active sites. Whereas when the inhibitor binds, it stabilizes the inactive form. So the enzymes are much more complicated. in their physiology than we think, right? Because if you can control enzymes, you can control the organism, right? Everything in the functioning of an organism is related to one or another enzymatic reaction. Can you see here? So the substrate is the yellow color that should be binding to here, no? It should be binding to the active site here, no? So this is the best shape, that is the active form to do the reaction. But what is the other form? Can you see? So what are these? These are subunits, no? Multi subunit enzyme. So this is the inactive form. It's useless, no? The substrate can't bind them. Right? He can't bind them because why? It's inactive. What has been done? This inhibitor has bound to the inactive form and it has stabilized. It doesn't allow it to change. It lock right? So that is what's happened in allosteric activation and inhibition. Simple thing, right? Any questions? Allosteric activation and inhibition. Okay. So what is the example? You have ADP. So in page number 57, you can see there's an example. ADP function as allosteric activator. Allosteric activator full stop bind to the enzyme and stimulate the production of ATP by catabolism. If the supply of ATP exceeds the demand, catabolism slows down as ATP bind to the same enzyme as inhibitor. Okay. So this story, ATP is produced by an enzyme called ADP. Then ADP plus pi is converted to ATP by an enzyme called ATP synthase. ATP synthase. So what needs to be done? I told you we can't store ATP. Ne? ATP we can't keep somewhere. ATP. So when we have too much ATP, we need to stop the production of ATP. How does it do is, this ATP is going to bind to the allosteric site of this ATP synthase and it's going to inhibit. So sorry, it's going to inhibit. ATP is going to inhibit ATP synthase. එයාම inhibit කරගන්නවා right මාවම හදන එක දැන් ඇති මම right there is too much of me, it's wasteful, stop කියලා කියනවා. So ATP itself inhibits itself. Then the other way around, if you have too much ADP, what does that mean? That means the cell doesn't have enough energy, no? ATP මදි කියන එක. So if there is too much ADP, that is going to activate. Write this down under regulation. Write a small topic under this diagram here. Write down allosteric regulation of ATP synthase. Write down and allosteric regulation of ATP synthase. Allosteric regulation of ATP synthase. And write down this reaction. ADP plus pi is converted to ATP by ATP synthase. ATP itself is going to inhibit ATP synthase, but ADP can activate ATP synthase.
Can you see this is how this ATP synthase enzymes looks like? Okay, it's a very complicated enzyme. From Huawei, it's like a turbine, no? Right? It's a turbine that is going to produce ATP. මෙතන තමයි ඔය ඔය story එක වෙන්නේ. When there is too much of this ATP, it's going to inhibit this process. Right? When there is too much of ADP, it's going to activate this process. Allosteric activation. Anyway, there are better examples than this to explain this, but it's what is in your resource, right? We'll come across this ATP synthase a lot when you're doing photosynthesis and cellular respiration. Okay, so let's do cooperativity and feedback inhibition next week. Okay, write down, take out the essay writing book, write down the homework. Write down the homework topic. We are writing a full essay, 1A. Briefly describe. Online people, write down. So, any new students, I need you to have a separate essay writing book, okay? CR book to write essays. So, topic part A, briefly describe the general characteristics of enzymes. Briefly describe the general characteristics of enzymes. Part B, 1 and 2 කියලා දෙකක් තියෙනවා. Okay, part B1, explain how pH and temperature, affect the rate of enzymatic reactions. B2, want me to read it again? Explain how pH and temperature affect the rate of enzymatic reactions. B2, explain the action of competitive and non-competitive inhibitors in enzymatic reactions. Explain the action of competitive and non-competitive inhibitors in enzymatic reactions. So, I will give you a hint about the marking scheme. This should come up to a total of 40 points, 40 possible points. that will be multiplied by 40, yeah, multiplied by 4 actually, but any maximum of 36 will give you the full marks. Okay, there are 40 possible points, but any 36 is going to give you the full marks, multiplied by 4, and balance you have to guess, right? Okay, there should be some diagrams drawn, right? At this point you understand this, right? Whenever an essay topic is given, first you have to consider if this topic requires a diagram, right? Because definitely that is going to carry marks in the paper, right, in the marking scheme. So this topic requires some diagrams, so focus on those, study the section using the resource book, close the resource book and write the essay. We'll discuss the essay next week, and also I'll try to demonstrate the practicals next week as well, quickly. Okay, quickly, otherwise boru. So as you can see, we covered a lot today. We covered a lot, but there's a small section remaining, cooperativity and feedback inhibition. Quickly we'll cover. Any questions, if you can, online people or anyone, you can ask, right? Um, if you need additional information, send a message to this number. In a practical report, what do you mean in a practical report writing? Usually you don't get that in the paper, right? You need to include the theory and the process and then observations. Those are the three parts of any practical, right? There's a theory, procedure and the observation. So, ituna thamai thiyenne ඕනම practical report එක. You don't need to write practical reports at A level, right? Okay. If you don't have any questions, you can go. Thank you for coming. Go safe. So, what's your plan? System thamaya. Okay, online people. Oh, come on. Okay, thank you.